wetlands, and including the paddy fields which we have been cultivating are all treated as wetlands in the international wetland definition. And these wetlands are 42 Ramsar sites have been identified in India. A Ramsar site is a wetland site designated of international importance under the Ramsar Convention. The Convention on Wetlands, known as Ramsar Convention, is an intergovernmental environmental treaty established in 1971 by UNESCO and coming into force from 1975. If one of the wetlands, these are the 42 wetlands that have been designated so far in our country. Very few in our south, but there are 3 in Kerala and 1 in Tamil Nadu, 1 in Andhra Pradesh. So the rest are all mostly in the northern part of India. Just to give an example of Kolleru Lake in Andhra Pradesh, Nellore district, this was completely devastated by the aquaculture for prawns and fishes, and that has been warned by the UNESCO to restore these particular wetland, and all the government of Andhra Pradesh has taken interest and then restored this particular wetland, because more than a crore of people are dependent on this particular wetland for their livelihood. And apart from this, large number of animal species which are dependent on this lake for their existence. And this is a very important wetland in the one of the important wetlands in the south. Another wetland which I will give you an example is a wetland in Manipur, which is very important for what is called as this is Kaibul Lamjo National Park, which is there in Manipur. And what you see here are the islands of grasses, the islands formed by the mat of grasses. And these grasses, they are the floating islands almost. And you can see some of the people live on this. And this grass hosts one of the unique deers called Sangai deer, which is endemic to this particular, and this is several kilometers long. I will not go into the details, just to give an idea as to how important these wetlands are for the local people. Then coming to coastal and marine ecosystems, the mangroves, most important mangroves, 14% of geographical area in India is covered by mangroves. 7% of the world's mangroves are in India, and the most important are the 2 important deltaic regions, the Mahanadi Delta and also the Sundarban Delta, where we have the largest amount of mangroves in the world. And then other important, the coastal marine ecosystems are the sea grasses, which we have only 14 species, seaweeds 950 species, that include the higher algae or the macroalgae. Then coming to desert ecosystems, hot deserts and cold deserts. Hot deserts, 2% of the India's landmass. 2% of the India's landmass covers hot desert. 2% of the India's landmass, Rajasthan, Gujarat, Punjab, and Haryana. Cold desert, as Jammu Kashmir is distributed in Jammu Kashmir, Himachal Pradesh at altitudes 4,000 to 6,000 meters, where less than the temperature goes down to minus 45 degree temperature, and 10 to 70 millimeters rainfall. And this is the kind of diversity of ecosystems that we have in our country, which sustains and supports immense biodiversity. So just to give you an idea as to the forest ecosystems, the major types are the tropical forest, subtropical forest, temperate forest, subalpine forest. And these 4 types are divided into 16 major types. And of all, the tropical dry deciduous forest covers about 38% of the India's geographical area. The next tropical moist deciduous forest, which I showed the pictures in the initially, covers about 30% of the particularly in the central and eastern India. This is the situation of forest in our country today, and very alarming situation. In fact, we have lost most of the forests, and we have been struggling to maintain even at 24% of the land area or the geographical area of our country. And only some forests are left in the Western Ghats and Eastern India and Northeastern India and Andaman Nicobar Islands. So we need to increase the forest cover, and efforts are to be made by all the forest departments, respective state departments, to increase the forest cover to reach the international requirement of 33.33%. I will not go into the details here. Now, the Botanical Survey has been was formed in 1890 and reorganized in 1984, and it has 7 regional centers located in different phytogeographical regions with headquarters at Calcutta. These details I will give you much later in my second lecture. And so far, we have covered about nearly 75% of the the country is fully explored, and 40 to 70% is explored. I mean, the purple mark that you see here, we have covered only 40 to 70% to be explored, and less than 40% is we have not been able to cover less than 40% in many of these areas. Coming to the forest diversity that we have documented so far, you see, I have covered all the groups here, including bacteria and viruses, for simple reason. We have in Botanical Survey of India, we have angiosperms, gymnosperms, pteridophytes, bryophytes, algae, lichenize fungi, and fungi. Bacteria and viruses, although we do not work, but our scientists have been encouraging, documenting based on the published resources, and then giving the figures here. And here you can see that I have given a figure December 2019. December 2019 is the time up to which this whatever the documentation that has been done is recorded here is given up to figures given up to 2019. Coming to angiosperms, in the whole world we have about 416 families of angiosperms, of which 251 are represented in our country, and we so far 3,70,000 species under 13,500 genera are documented in the world. In India, 18,800 species under 3,060 genera are documented, which comes to about 5% of the angiosperms of the world are represented in our country. Coming to gymnosperms, 1,090 species are documented in the world, and about 82 species are there in our country. In addition to this, we have some more the cultivated species which are introduced as ornamentals, and that represents about nearly 7.5% of the diversity that we have in the world. Pteridophytes, 13,300 species, of which we have 1,307 species documented. And this group we have fairly documented, including gymnosperms, and has about 9.82% in our country. But again, let me tell you, pteridophytes being, they produce a large number of hybrids, and therefore there is a need for a critical study of many of these ferns and fernalies for these, their correct circumscription of the species, correct validity of the species, correct identification of the species. So we have fairly good number of pteridologists who can really undertake this work, and there is a need for immediately undertaking the completion of the pteridophytic flora of our country, and there should be a coordinated effort in this particular area. Coming to bryophytes, again we have very limited people for bryophytes, now not more than about 10 people across the country, and most of these species, so far 20,300 species known in the world, 2,786 are documented in our country. But again, very limited number of people are there to document these resources. And most of these documentation is done from the Northeast India and areas which can be easily accessed by people, or I would say the tourist important areas where people easily can go and collect these areas. In Western Ghats that has been done, but this is most insufficient. We need again a large number of people to work out this particular group, and which has so far documented about nearly 14% of the world's debris in our country. And algae is a unique group which has both these freshwater, marine, and terrestrial, and also even epiphytic algae are present. And these, so far in the world, we have about 44,000 species. In our country, we have 7,434. Again, this is a very, very, very minimal. So we need to, as far as the marine macroalgae are concerned, we are fairly good in our documentation, but there are microalgae in the oceans and also in the freshwater bodies and other water bodies in our country, which needs a large number of algologists to document these resources. And so far, we have documented only 17%, nearly 17% of the algae in various habitats that we have. Then coming to fungi, before that, I called the lichens as lichenized fungi because the recent classification based on molecular studies showed that these are more related to fungi, although it has partners of both algae and fungi both together in a symbiotic relation. And so far, 19,387 species have been documented in the world. Of course, again, whatever figures I am giving here for the global figures that I am giving is already about 4 years old. So in between, a large number of species have been added, new species have been added, and these new species are not being taken into account here because there is no compilation on a global basis after 2017. So you have to bear with me for the figures that I am giving. You can always add the plus or minus say nearly 1,000 species across all the groups. And I would say that these lichenized fungi in India, we have again 4 or 5 people, and those people are located mostly in NBRI Lucknow and BSI Allahabad and Andaman Nicobar Islands. So these are very limited people. In the university system, hardly I see only 1 or 2 people working in Andhra Pradesh and also in Maharashtra, these on lichens, but very limited number of people. But the diversity more than what we have actually documented so far. That comes to about 15%, but again, this 15% is also quite high when compared to the global figures. The global figures should have been much higher than 19,387. And I think once the documentation starts, particularly in the Amazon forest and African forest, and also in the Southeast Asian forest, the figures will go enormously higher. Coming to fungi, which is the largest group, 75,000 species have been documented. And in India, we have about 15,447 species, which comes to about 20% of the total world. Bacteria and viruses. These bacteria, viruses, fungi, mostly our people started working on these groups which affect the crops, cultivated crops, humans, and also the animals, which affect the, which cause diseases in all these 3 major biological systems. And bacteria and viruses, about 25,000 species have been discovered, and now we have added coronavirus to this, and 1,239 species in India, and that comes to about nearly 5%. That is again a minimal, and it is only a tip of the iceberg we have shown here, and the diversity of these bacteria and viruses is much higher than all the groups of plants put together. And I would say that this is one of the largest groups that which needs a separate survey, like botanical survey, zoological survey. We should have a microbiological survey and also document this. You need army of people in this particular area. If you look at the overall diversity in the documented in the world is about 5,68,077 species. In India, we have nearly about little more than 50,000 species, which comes to about 9.36% of the global biological diversity, particularly plant diversity. So some of the major groups like Orchidaceae, for example, 736 genera and 28,000 species in the world. And we have about 1,350 species in our country. And Asteraceae, 1,623 genera and 24,700 species. If you compare these two families, which are the largest families in the world, you look at the Orchidaceae, which has 736 genera, less number of genera. Asteraceae has 1,623 genera, so more genera here. But if you come to species, Orchidaceae has a larger number of species, 28,000, whereas Asteraceae has 24,700 species. So the entire diversity in our country, if you take both animals and plants, fungi, algae plus microbes coming together, it will come to about 1,52,173 documented up to December 2019. Why I am giving this figure December 2019? So I will tell you a little later. Now, if you look at group-wise, if you look at the group-wise disposition of our plant diversity, angiosperms 37.6. This is the largest. We have the largest number of people for angiosperms, and therefore our documentation is almost nearing completion for the angiosperms are concerned. So that is about 37.60%, and gymnosperms 82.16%, pteridophytes 1,307, 2.61%, and likewise bryophytes, algae, lichenids, fungi, bacteria, and viruses. You can see out of 50,000 species, you can see the share of various different groups that we are having in our country. So in only one family was described as Brachycalaceae, which was considered endemic to Sikkim, but later on it has been found that based on the molecular studies, this Brachycalaceae, monotypic, means with a single species and genus, has been merged with Rosaceae based on the molecular studies, and it is no more endemic with us. Then other new family records that have been made are Hydrotellaceae, Cystaceae, Aconiaceae, Petrosaviaceae. So these additions have been made after the flora of British India, or the new flora of India, in fact. So these species are based on, you see, if you look at, only one example I will give, Hydrotellaceae, Trichuria concatensis, a new species and a new generic record for India and a new family record for India. Its nearest relatives are there in Tasmania and New Zealand. Cystaceae, this is understandable because desert continues into the Thar desert in western region of country, particularly Rajasthan and Gujarat, where this Helianthemum lippi grows in Pakistan, but it has been recently recorded from Gujarat, recently in the sense 2016 already. Aconiaceae, which occurs in China, which has been documented from Arunachal Pradesh, and similarly Petrosaviaceae, another family, Petrosavia sakuria, which is documented from Arunachal Pradesh, Pale Valley, which occurs in China, adjoining China, Indonesia, Japan, Myanmar, and Taiwan, Vietnam. So this is the Trichuria concatensis, which looks like a moss plant. I will not go into the details, but it's you need a special eye to even dissect and study this particular species. And this is Helianthemum lippi, which is documented from the Western Ghats of the Western India, particularly the Gujarat region. And these are the flowers of Helianthemum lippi. Yeah, the whole plant of Helianthus lippi. This is Aconiaceae, the Betsnidiera sinensis, which is documented from Arunachal Pradesh. The Petrosaviaceae, Petrosavia sakuriae, which is also which is which is recorded from Arunachal Pradesh again. And if you look at the most important thing, Convention on Biological Diversity has given us the sovereign rights of all the resources that we have in our country or in the respective countries in the world who are signatory to the Convention on Biological Diversity. If you look at this particular table, which gives you the endemism in each one of those groups, if you take angiosperms, out of 18,800 species, 4,403 species are endemic to our country, and that comes to about 23.42%. Gymnosperms about 14.63%. 12 gymnosperms are endemic to our country. Like most important, I will mention little later. Pteridophytes, 235 species. Bryophytes, 650 species. Algae, 1950 species. Lichens, lichenized fungi, 553 species. Fungi, 3,635 species. Bacteria and viruses, 110 species that we have documented. Why I am mentioning these particular endemic plants is very, very important. Any development, any drug that we develop or any economic product that we develop out of any of these species, since they are endemic to our country, and these are these will fetch large amount of income to the government, because these are found only in India. Even if they are taken to other countries, and if they produce some product out of this, they need to share the benefit of 50 to 50-50 with our country, and therefore these endemic species are very, very, very important, and these are the most priority species even in conservation, because they occur only in India and nowhere else in the world. If you look at in the Himalayas, more than 3,600 species are endemic to Himalayas. 250 species are endemic to Andaman Nicobar Islands. 3 mega centers of endemism: Eastern Himalaya, Western Himalaya, Western Ghats. 25 microcenters which are distributed across our country. And 139 described plant species are known to become extinct. You can imagine the kind of see, the BSI has documented, brought out what is called as the endemic vascular plants of India, and this gives you all the species that have been documented up to that date. And then we have the dominant of the 15 families, the dominant family is Poaceae with about 335 species. Orchidaceae with 274 species. Similarly, Leguminosae with 274 species. I will not go into the details of all these families, and I will share with these slides. All of you can leisurely read. And if you look at the dicotyledons, Leguminosae has, again I mentioned, 274 species. The next family, largest family, is 235 species. Poaceae, Poaceae with 335 species, Orchidaceae 274 species. Among the states, Tamil Nadu tops with 410 species of endemic species. You can imagine Tamil Nadu is, although we always think that Kerala has the very thick forests and diverse endemics there. And if you compare the Kerala, Tamil Nadu, Maharashtra, Karnataka, and Tamil Nadu has the highest number of endemics in our country. I mentioned the figure December 2019. This figure I mentioned because every year the Botanical Survey of India, Zoological Survey of India brings out what are called as the plant discoveries and animal discoveries. See, for the 2020, we have brought out the plant discoveries. And the 2020 plant discoveries that is published is from the species that are recorded for the current calendar year 2019 are published in 2020 and then released on World Environment Day on June 5th by the minister. So here, Babul Supriyo, the Minister for Environment and Deputy Minister for Environment and Forest, has released the last year's plant discoveries, which he is flanked by the Director Botanical Survey of India, Director Zoological Survey of India, and that is the reason why I have been mentioning the figures that I give in my presentation are up to December 2019, and 2020 figures will be added now because now this is being compiled and it will be released for 2020 figures. The plant discoveries will be released on the June 5th World Environment Day by the minister again. And this is the 2019 plant discoveries, which was released in 2020, and 2020 will be released now. And you can see here, one new family has been added, 42 new genera, 1,719 new species have been added during the year. So these plant discoveries that are documented here covers not only the new species discovered by the Botanical Survey of India scientists, but also scientists from all over the country and all over the world. All over the world in the sense, those species which are described based on the Indian material and are found in India are included from outside the country also. Now, these endemic species, I said that there are many endemic species, say for example, flowering plants, more than 4,400 species, maybe now it will be 4,500 species, including the new species that have been described in the last 1 year. And these species are very unique. And I said, these are found only in our country and nowhere else in the world, and therefore very, very important. Now, if you look at the ornamental species that we have in our gardens and parks and wherever we go, in even our kitchen gardens, and all these species are mostly introduced from outside the country. We have large number of very attractive ornamental species in our country, and there has been no attempt to document, to not only document, but also to domesticate these species and develop package of practices for cultivation of these species. You see, this is, the previous one is Alicicarpus belgamensis, endemic to Belgaum in the Karnataka and Maharashtra. This is Cyanotis concanensis, endemic to Maharashtra. This is the Smithia hirsuta. I mentioned the Khas plateau or the plateau of flowers, and this is one of the dominant species there, and which imparts color to the entire when it comes to flowering. And these are some of the Murdanias. These are the Ceropegias. Again, the Ceropegia occulta, Ceropegia jaini, and this is Indigofera, which is again very attractive species in the plateaus. I'm just taken a few plateau flowers here, and this is Pinda concanensis. For a long time, it was, since its description, I would say, it was endemic, it was a monotypic, but recently another species has been described also from Maharashtra. And this is how a large number of, Musa species are described from northeastern region, and these wild germplasm resource is very, very important for the improvement of the banana and the banana research station, particularly which is located in the Tiruchirappalli. And on the left hand side, you see Delphinia malabaricum. This particular species, this is the only species an attempt has been made, not by even the horticulture scientist, but by a botanical scientist, Professor S.R. Yadav from Kolhapur, who has attempted to domesticate this particular species. It is a wild species which commonly occurs in the Western Ghats, but this has been domesticated now, and they have developed a package of practice, and there is a demand for the cut flowers of this. But again, there is no popularization of this developed or domesticated uh, wild ornamental plant. And similarly, if you look at on the right hand side is the Pogostemon deccanensis, which is again a very common and gives you, imparts color to the paddy fields after harvesting, particularly in the lateritic areas of northern Western Ghats. And this is what I want to say. But the most important thing is taxonomy. We need more people to come undertake the studies, and to document our resources, we need a large number of taxonomists, and we need to train taxonomists. And the Government of India has started a program called the All India Coordinated Project and Capacity Building in Taxonomy in India. And it had the entire project had a checkered history, and more people are required. But at the same time, the governments, state governments, as well as the central government has to create the job opportunities for most of these students. Unless we create job opportunities for these taxonomists, we will not be able to attract the taxonomists to undertake the floristic surveys and documentation of our floras, and later identify and conserve the flora, and also identify species which need sustainable utilization and all that. So thank you very much. And if you have any questions, I will be happy to answer.
Thank you, sir, for such an impactful lecture on flora, the significance of Indian floral diversity, and the importance of their conservation. Now, the participants can clarify their doubts. Don't hesitate to ask your doubt, guys. Feel free to ask. If you guys have any doubt, you can ask here.
No, I always say at the end of my lecture, either people have understood everything that I have said and they have no doubts, or they have not understood anything out of this. So please clarify or ask anything that you want clarification on about this floristic diversity. It should not be one-way traffic.
Good morning, sir.
Good morning.
Sir, you mentioned about the endemic species that from which the drugs are being extracted, and that would fetch a lot of income to our government.
Yes.
And you also added the importance that these species are found only in India and it's not found in any part of the world. So are there any massive actions or organization or any committee being created in order to restore them, sir? That is by the government. Any act is being taken, sir?
Restore them for what? Actually, there are, as I mentioned in my this thing, that these are the endemic species which are found only in India. Not all species, only endemic species which I mentioned in one of the tables. I mentioned that the percentage of endemism. For example, under angiosperms, 4,400 species are endemic to our country, and they are distributed throughout the country with maximum number of species, about 3,600 species in Himalayas alone. And these species, there are many, like I will give just one example. Say Mapia fetida, or even Taxus wallichiana. Taxus wallichiana yields Taxol, a cancer-treating drug. And similarly, Mapia fetida yields drug for treating cancers. So therefore, these particular species are being exploited in such a large scale that the species are threatened with extinction. But now they have found that the endophytic bacteria in Taxol, which produces Taxol in Taxus baccata or Taxus wallichiana, now there is exploitation has come down because the cultures are being made of this endophytic bacteria and from which they produce Taxol. Similarly, there are many other drug species which yield drugs. For example, even if you take Saraka Ashoka, which is a very important Ayurvedic plant, and this species is threatened in the nature, which is not really endemic, but occurs in only in Eastern Ghats and Western Ghats and Northeast India, and this is being most sought after by the herbal industries for the production of Ashoka ristum. So there are many such endemic species which are not only being controlled by the forest departments, but also by the international market. Like there is what is called as CITES, Conventional International Trade in Endangered Species of Flora and Fauna. So there are controls or export of any of these species or their parts. But if the finished products are produced here, they can be exported. So I don't know whether I have made your point clear or not. If you have any further questioning, you can ask me. Hello?
Yes, sir, I got clarified.
Ah, thank you.
Participants, do you have any other doubt? You can clarify it with Sanjapa sir.
Sir?
Okay, sir.
Yeah.
Sir,
can
how
we
was
have
your Silent Valley experience, sir?
Silent Valley is now, it is silent. Otherwise, it was burning once upon a time. In the sense, they wanted to construct a big dam there, hydroelectric dam that has been shelved under the intervention of Government of India. And now it is quite well preserved and well taken care of by the Government of State, Forest Department of Kerala. Well protected and the diversity of the species are quite well protected because it is part of the national park. It is part of the Nilgiri Biosphere Reserve.
Thank you, sir.
If you want to know the diversity of species, there is a flora written by Silent Valley flora written by Professor A.K.S. Manilal from Calicut University. You can refer to that, which gives you the diversity of species. There are many more species that have been described after this particular flora, but to get an idea as to what is the kind of diversity, you can refer to that flora.
Yes, sir. Thank you, sir.
Thank you for clarifying the doubts and for this wonderful lecture, sir. Thank you so much.
Thank you. Thank you.
Sir, can we continue with the next session or shall we take a break for 5 minutes, sir?
You take a break for 5 minutes and next lecture is by Dr. S.R. I don't know.
Yes, sir. Yes, sir.
Good.
Yes. So participants, you can have a break for 5 minutes and please join us after that. Thank you.
Ma'am, I have one clarification.
Yeah.
Sure,
please.
sir. You can ask.
Hello, ma'am.
Yeah.
Hello, sir. I am Habib Rahman from Jammu College.
Please.
The wonderful presentation and beautiful images. And how many species are identified from the Western Ghats region, cyanobacteria and lichen species? Sir?
What? I am not able to get your point clearly.
Western Ghats, how many species was identified from the cyanobacteria and algal species sir, at Western Ghats region?
nobody has compiled any
Yeah.
number of algal species from the Western Ghats so far.
Yes, sir. All the species are lichens or cyanobacteria.
No,
How
no.
is it,
Only
uh,
angiosperms and gymnosperms have been identified clearly, compiled nicely. But algae, fungi, bryophytes, pteridophytes, these are not identified for across the entire Western Ghats. Yes, Western Ghats pass through, 6 states.
okay, sir. How is it work it possible at this region, sir?
Huh?
Sir, how is it the work is possible in this region?
You-
Cyanobacteria and lichens.
No, you can undertake. See, as I mentioned, when it comes to lichens, bryophytes, people have visited, easily accessible areas wherever, vehicles are possible to go. Because interior, if you take, for example, Silent Valley interior, you need permissions from the state forest departments, and also you need to stay inside the forest for a longer time to survey all these areas. And unless that is done, we will not be able to document the entire diversity. So there were chances for documenting, particularly the angiosperms, these larger species, but smaller species like algae, fungi, bryophytes, pteridophytes, and all,
Cyanobacteria.
they take very long time, and we don't
Okay.
have many people to work on.
Okay. Okay.
So
Thank you, sir.
if you have interest, you should start undertaking maybe nearest place to you, maybe Kodaikanal or somewhere. If you
Yes,
undertake,
sir.
then it will be a wonderful idea. Even Botany Survey of India has not been able to cover the entire Western Ghats for all the groups except for the angiosperms. And
Okay. Okay, sir. Thank you, sir.
oh,
Hello, good afternoon, sir.
संजप्पा सर
ah,
संजप्पा
Hello.
सर नमस्कार
Namaskar. Oh, Yadav Sahab.
हाँ यस सर
No, some clarifications that people are seeking, we can talk after your lecture.
ओके
Okay.
ओके
The next question, please. Hello.
हेलो सर, मैं गुजरात से हूँ। फेबेसी फैमिली में एक प्लांट है टेरामनस, उसमें टेरामनस लेबियालिस और टेरामनस मोलिस, एक स्पीसीज टेरामनस रिपेंस जो महाराष्ट्र में मिलती है, उसके बारे में सर कुछ डिटेल्स बताइए ना।
डिटेल्स हाफ एंड तो नहीं बता सकता हूँ, लेकिन आप जो तीनों स्पीशीज बता रहे, कौन सी एरिया में है, या डांस में या कहाँ आप कलेक्ट किया उसको?
नहीं सर, लेबियालिस और मोलिस में बहुत ही वेरिएशन रहता है, लेबियालिस और मोलिस में। लेकिन रिपेंस का बहुत डिटेल्स नहीं दिया है। उसके जो पोड्स है और सीड के ऊपर से उसको रिपेंस अलग किया गया है वेराइटी से। वो गुजरात में लेबियालिस और मोलिस दोनों मिलते हैं।
हाँ, मैं राजपीपला में कलेक्ट किया, जाम्बूगढ़ में कलेक्ट किया, नियर बड़ौदा।
यस सर, मैंने वांसदा से किया है, डांग से लेबियालिस।
हाँ, लेबियालिस डांग के बहुत कॉमन स्पीशीज है वो तो।
यस सर, दोनों पूरे गुजरात में मिलते हैं, लेकिन रिपेंस के बारे में थोड़ा जानना था क्योंकि वो बहुत ही वेरिएशन वाले स्पीशीज है। और
आप मुझे वो उसको इमेजेस भेजिए, नहीं तो स्पेसिमेन भेजिए, मैं आपको डिटेल्स देंगे उसका।
ओके श्योर सर, थैंक यू सो मच।
मेरा वो ईमेल एड्रेस सब नोट कर लीजिएगा। All of you note down my email ID so that you can, any doubt, any identification help that you require, you can always send it to me, and I will try to help you in identifications.
Sir, I think Dr. C. Divya, she has sent you in plant image and she's asking you the name of the plant, sir, in the chat box.
Chat box. I don't
Yes,
know.
sir. So down there is an image chat. If you click that, the chat box will open, sir.
Image chart, no, I don't get this chat box here.
Sir, near to share screen, there is a image.
Oh,
This
chat.
icon, sir.
Yeah.
Yes, sir. If you click that, the chat box will open.
Chat box has opened. Which plant, Divya? Image is here. Yeah,
Yes, sir.
let me save that image. Because image should open, no?
Yes, sir. Yes, sir.
Mhm. This. I am not able to.
Okay, sir.
You send
So if
me.
you
I'm not able to because I am not computer savvy.
okay, sir. So if you just click that image, I think it will get downloaded.
No, it is going for download.
Yes, sir.
Sir, after the image has got downloaded, you just double click on the image, sir, on the chat box. That is getting opened.
That I know, but the image is taking a lot of time. Wait. I have no idea. This I cannot tell you what exactly is this. I need to. I have no idea about this plant. This may be some Scrophulariaceae member. It looks like. Hello.
Yes, sir. Divya, see Divya, are you there in the session?
Yes, yes.
Yes.
No, you see the
Three images.
any image. It is very difficult to describe. I mean, identify based on images most of the times, unless one has worked in that particular group. So, if legumes to some extent, I can because I have been working on legumes. So this looks like a Scrophulariaceae member. You spend time and send your email ID so that I will send the identification.
Okay, sir. Images to email ID, sir.
You note down my email, sanjappam@ymail.com.
Sir, 1 second, please.
Sanjappam, SANJAPPAM @ymail.com. Ymail, not Gmail.
Okay, sir. Okay, thank you, sir.
So you send it to me. I will just try, and if not possible, and I will ask some other people also, because I will not be able to identify all the species of all the families. So I have concentrated my entire career on few families which I can easily identify if they are from India. Any other question?
Sir, shall we start with the next session?
Yeah,
Yes,
you
sir.
asked Dr. Yadav if he is ready, I think.
Yes, sir. Yes, sir.
Yeah. Thank you.
Thank you so much, sir.
Okay.
Now, I kindly request Dr. M. Kamalam, Associate Professor, Department of Botany, PSGR Krishnamal College for Women, to welcome Speaker of the Technical Session 2, Dr. Yasar Yadav.
Good morning to one and all present here. I am humbled in welcoming our resource person, Professor S.R. Yadav, for the Science Academic Virtual Lecture Workshop. On behalf of Botany Department and PHRKC, I welcome you, sir. I'm also happy to see Professor Sanjapa here. Thank you, sir, for organizing this wonderful virtual workshop in this pandemic situation for the benefit of the students. Thank you, sir. I feel proud and privileged to welcome Professor S.R. Yadav, a versatile personality, and his journey in the field of academic and research is very long. Due to some constraints, time constraints, let me brief his expertise. He obtained his degree from Mumbai University and started his teaching journey from 1978 and is still continuing in it. He has served in several colleges, universities like Goa, Delhi, and is now working as an INSA senior scientist in Shivaji University. Dr. Yadav is an outstanding plant taxonomist of international repute, an excellent teacher who is floristic and taxonomic works, particularly flora of Kolhapur and grasses of Maharashtra, have tremendously influenced many students to take up plant taxonomy as their career. During the last 3 decades, he not only recorded a new flowering plant family, Hydrothalaceae, but also discovered 74 species new to science. His contribution on cytotaxonomy and biosystematic studies of Chlorophytum, Borleria, Therapogia, Dremia, Leptoboria, Crinum, Aponogita have been widely acclaimed. He is an ardent field botanist and outstanding plant photographer. His works on ex situ conservation, species recovery and rehabilitation of nearly extinct species, and Hubbardea, Heptanero, and Serapogia fantastica, and the establishment of a lead botanical garden are exemplary. Beyond this, to his merit, he has guided 11 MPhil students and 26 PhD scholars. He has published over 270 research papers in national and international journals, and he has published 8 books and contributed chapters in more than 10 books. He has completed over 20 research projects. He won several awards, and the most prestigious one was the Best Teacher Award. As a resource person, he has visited to Japan, China, USA, Iran, Australia, Nepal, and Brazil, and the list is going on. Having said all, I now request the renowned personality, Professor S.R. Yadav, to share his expertise to the future India waiting in front of him in online. Thank you, sir. Thank you once again. I welcome you to chair this session.
Thank you, ma'am. Now I call upon Dr. S.R. Yadav, Professor, Department of Botany, Shivaji University, Kolhapur, to deliver his lecture on the topic Biosystematics, a tool to understand microevolution, diversification, and speciation in plants. The session is over to you, sir.
Yadav, sir.
Hello? Hello? Hello?
Yes, sir. We hear you. Yes, sir.
I am facing some problem in internet, but now I will share screen. Is it visible to all of you? My screen. Hello?
Wait a minute. Yes,
Hello?
yes, it is. Yes, it is visible.
Hello?
Sir, it's visible, sir. Your screen is visible.
Screen is visible. Okay, good. Right. So, good morning to all of you. Just now, we have listened Dr. Sanjappa, a very detailed lecture on biodiversity or plant diversity of our country, various aspects of plant diversity of our country. And you all participant in the beginning only, I want to tell you something about what he said, what we have. We are one of the rich on the planet with reference to biodiversity. And especially plant diversity, reason is plant is the only biological capital of human being. All other capitals are of secondary importance. Plant capital or plant diversity is a real capital of human being. And he has made us aware of what is the diversity our country has. Today, my lecture is on a tool to understand microevolution, diversification, speciation, and in plants. Now, this usually people do not go for such a studies because it is a time consuming. It does not give many papers because people are hungry for publishing papers after papers and making their biodata thick. But reality, if you want to understand plant, we need to go for biosystematics. If you want to understand microevolution, we need to understand, plants. You have to go to the field and then only you can understand evolution, which is a very important aspect of our biology. Now, biosystematics. Really speaking, I do classical taxonomy, which is known as alpha taxonomy, going to field, collecting plants, studying morphology, and then, identifying them, describing them. That is our job. And second part comes, that is experimental taxonomy or biosystematics. We call it as omega taxonomy. In India, really speaking, very little work has been done on biosystematics because it is a lifetime work which gives you 1 or 2 paper after 10, 15 years of work. So nobody has that time to spend to study plants. But anyway, some efforts I have made with Indian examples. I will try to explain how it is very interesting for any biologist. So biosystematics is concerned with cytological, genetic, and ecological aspects of taxonomy. We are exploring our flora, documenting it. Do you know how many species, percentage of species is known cytologically from India? Only 40% plant species have recorded chromosome number. Remaining 50 to 60% species are yet to be studied, even only for chromosome number, not even karyotype. Genetics, ecological aspects, those are the really concerned with biosystematics. It involves studies in the field and experimental garden. You need to have experimental garden, then only we can do biosystematics. And biosystematic may therefore be considered as a taxonomic application of gene ecology. That is, study of the genotypic and phenotypic variations of the species in relation to environment in which they occur. Many times, many species are described which do not stand as a distinct species. We do find variations in different phytogeographical regions, in different zones of our country, but that species, whether it stands as a distinct species or not, is a very difficult question for anyone, and many paper species have been described which may not stand in future. And that's why whenever you describe some new species, you have to be very careful with reference to environment, its cytology, its ecology. Then only we can, you should dare to describe new species. Biosystematic is a branch of botany and is variously termed as experimental taxonomy, gynecology, genonomic, modern taxonomy, and new systematics. Similarly, what are the aims of biosystematic studies? To delimit the natural biological units. For a taxonomist, classical taxonomist, we have species, subspecies, variety, forma. These are the natural units we recognize, but whether below species are there units which we can delimit on the basis of cytogenetical studies. That is one part. Second, to apply these units to the task of conveying precise information regarding their defined limits, relationships, variability, and dynamic structure. Thus, the critical data from comparative morphology, cytology, genetics, and ecology, when applied to organic evolution, is known as biosystematics. Now, basic units in biosystematics. In modern population taxonomy, instead of regarding species as a basic unit, it is a local population that is taken to starting point, the evolutionary unit. In Western Ghat, we find many genera which have become epibiotic. They are actively evolving. If you really want to understand evolution, what is happening in Western Ghat, we have to see the epibiotic species which are evolving actively and then understand how they are evolving. Local breeding populations or Mendelian population has been defined as a reproductive community of sexual and cross-fertilizing individuals which share a common gene pool. Okay, so that is a Mendelian population. Biosystematic is revivified systematics that takes into account the genetics, the evolutionary nature of groups, and concern with microevolutionary phenomena. Okay, so how new species are formed? If you want to really understand, you have to study microevolution, which finally result into differentiation of species. Okay, so biosystematic uses the group concept based on reproductive isolation and genetic structure. Now, reproductive isolation is one very important aspect in defining species, and its genetic structure is equally important, but does not attempt to name these groups in the formal taxonomic units. We recognize units in biosystematics, but we have no formal names for those units. Biosystematic is regarded as a further extension of classical taxonomy. Methods in biosystematics, all of you know, this is a classical chapter in every textbook. Methods in classical taxonomy are structural information. Methods in classical cytology, chromosome information, chromosome structure, chromosome behavior. This you need to study. Methods in genetic information from breeding systems, the extent of hybridization. So this is the third aspect one has to do, and cultivation of those forms, units under uniform or very climatic environment needs to be done to understand the characteristics and their evolution. So in biosystematics, usually there are some other names also, but I will only define these 3-4 units: ecophene, ecotype, ecospecies, and synospecies. These are 4 categories recognized in biosystematics. Now, what is ecophen? An ecophen is constituted by individuals occupying a particular habitat and adapted to it phenotypically, but not genotypically. The group is capable of hybridizing with other such groups to give hybrids showing complete fertility. So here, what it is said in nutshell, this is a genotype which is adapted to particular climatic conditions. It hybridizes freely with such a related groups and produces fully fertile hybrids. So this is only a morphological variants which are adapted to that ecological condition. Those are known as ecophens or ecads. Then another is ecotype. Now ecotype, many ecotypes have been recognized. We can say that approximately equivalent to variety or pharma, but these variations which are there in that population, they are genetically fixed, not like ecads. They are genetically fixed, and therefore, even if you grow these ecotypes under same climatic conditions, edaphic conditions, they maintain the differences. That means the differences are maintained, and they are fixed by genes. So, but these ecotypes cross freely and produce fully fertile hybrids, and therefore, the genetic differences are not that vast, and we recognize them as ecotypes. This is the second. Then ecospecies can be defined as a group of individuals capable of hybridizing with one another to give fully fertile hybrids. They are capable of hybridizing with other groups to give hybrids showing some fertility. So ecospecies is a group of plants, group of individuals which hybridize with each other and fully fertile. But such other species, such other populations, it hybridizes, but does not, only those hybrids shows partial fertility. For example, species of same genus, they can hybridize with each other and show some degree of fertility. They are not fully fertile. And that is ecospecies, which is approximately equivalent to species in taxonomy unit. Taxonomy. And last one is individuals capable of hybridizing with one another to give hybrids showing some fertility from a species. This group is capable of hybridizing with other groups, but such hybrids are sterile. So it is approximately equivalent to genus. Intergeneric hybrids are difficult to make. And even if we hybridize, they are, sterile. Hybrids are sterile. Their populations do not continue. So those are species equivalent to approximately generic level. So these are 4 categories which are recognized in a biosystematic. Now I am going to take you to a very serious problem which you will understand when I discuss. So limitations of biosystematic. Studies applicable to generic or below generic level. We only study this at the generic or species level or variety level. But above that, tribal level or family level, we cannot apply biosystematics. Another very important, time-consuming and laborious work. And that is why nobody likes to go for this. But really, if you want to understand botany and plants, to understand microevolution, we need to study, at least to do some biosystematic studies, which will give you insight, right? Now, cytological characters of taxonomic importance. Cytology has, chromosomes, they are very important. They carry genetic material, and therefore, of course, now molecular studies have come up, but cytological studies are equally important. Number of chromosomes, morphology of chromosomes, behavior of chromosomes during meiosis, it gives a lot of idea about similarity in genetic material. Accessory chromosomes, B chromosomes, which are present in many plants. crossability, and species delimitation. So all these are cytological characters which we use in biosystematic studies. Now, cytotaxonomy, this is one branch and probably forgotten branch in our country today. Dr. Sanjapa used to do cytology long back. Now he does taxonomy. So he has done a lot of contribution in cytology. What is species? So before I go to further presentation, I want to ask one question to all the participants. What is species? Species, what is species? If I ask, none of you will be able to answer me this question satisfactorily. None of the definition is sufficient to explain what is species. Taxonomy is defined in their own way. Cytologists define species in a different way. So everybody has its own definition. And remember, species is a concept. I will call some population as a variety. Some other taxonomist will come and say, no, this does not stand as a variety. It should be raised to species level. And this goes on up and down. So species is a concept and it is a product of each individual's judgment. Therefore, it differs and everybody has a different concept about species. So this is the one definition. And it is finally said, there are so many definitions, none of them is satisfactory. It is said that if a good taxonomist, a good word is very important. A good taxonomist, if he says it is a species, it has to be considered as a distinct species. But it is a good taxonomist who has a good field experience. Number, they represent an important level of integration in living nature. They represent an important level of integration in living nature. In natural conditions, they maintain their individuality, and that's why they represent an important level of integration. This recognition is a fundamental to pure biology, and an inventory of species of animals and plants of the world is the baseline of further research in biology. That Dr. Sanjapa has already traced this point. This becomes basis for everything. Your list of plants, checklist of plants become basis for everything, for every further development in biology. And that's why that is very important. Every biologist, whoever he may be, molecular biologist or whatever, evolutionary biologist, he works with species only. And that's why species is a very important entity in biological studies. Species definition, Linnaeus. Linnaeus believed in constancy and sharp delimitation of species. Darwin believed in completely opposite view, variations and overlapping and ever-changing pattern of species. Darwin found it impossible to delimit species and concluded that in determining whether a form should be ranked as a species or a variety, the opinion of naturalists having sound judgment and wide experience seems the only guide to follow. This is Darwin's statement, right? And from that, you can understand how it is difficult to define species. Species concept. There are several concepts of species, of which typological species concept or morphological species concept, which we follow, taxonomists follow. And another is a biological concept, most accepted definition, the reproductively isolated system of breeding populations are very important to understand species. So this is, even that definition is not complete. There are some apomictic species which we recognize as a distinct, and apomicts, in the biological concept, we cannot define them. Group of interbreeding natural populations that are reproductively isolated from each other is a definition given. Okay. Now, cytological data offers significant insight to understanding of species concept in various taxonomic groups. Cytological data is valuable in delimiting taxa at various levels. It is one of the widely known cytological parameters for a very large number of plant taxonomies. And I am going to give you, now explain you some of the very simple examples from our country and how we have made mess in delimiting species. Now some examples I am going to give: Ledboria, Drimia, Dipcadi, Iphigenia, Aponogeton, Chlorophytum, Glypoclova, Pancratium, Amorphopalus, and so on. Now you see, Ledboria, now, with first monsoon rains, you will find many plants on grounds, open grounds, all around in Tamil Nadu, in Maharashtra, everywhere. You find one plant, we used to identify it as a Sila. Okay, so now it is a Ledboria, which has 59 species worldwide. Although Ledboria was described from India, it has been treated as a synonym of Sila in most Indian literature. In India, the genus consists of 4 ill-defined species. I say ill-defined. One is Ledboria viridis. Another one is Ledboria karnatakensis, Ledboria hyderabadenssis, Ledboria rivulata. Now these are 4 species I have shown. And Ledboria rivulata is a widespread species which shows significant morphological variations, morphotypes, and somatic chromosome number or cytotypes in different habitats. If you take survey and critically analyze, and even molecular studies have been done by some, and they have also come to same conclusion what we have come to conclusion. So that is the variation even under cultivation, range of variations, cytomorphology, and molecular studies support an occurrence of a single species in peninsular India. I am showing you some with photographs. Now see this Ledboria. Bulbs imbricated, racemes with numerous flowers. Numerous means 40. And pedicels 0.4 to 1 centimeter, filiform, bracts, biped, ovule 1 per locule, Sila hyacinthiana. So far I have not seen Sila hyacinthiana. Okay, it is a rivulata only. Bulb tunicated. Now, in both hyacinthiana and this one, bulbs are tunicated. How these terms have been used wrongly, I don't know. All Silas, Ledboria species have tunicated bulbs. Another writes Sinanthus and Histranthus. Now, this difference also does not stand. Okay, so we have Poincare, we have Viridis, and that way many species are being were described. Now see, Physia Poincare, Ledboria rivulata. Now Ledboria rivulata, as per our opinion is concerned, it is same as Sila hyacinthiana, Sila viridis salib, then Ledboria viridis, Ledboria junnarensis, Ledboria karnatakensis, Ledboria hyderabadens. Now you tell me, definitely anybody will get confused what is right and what is wrong, what taxonomists are doing. I will show you some photographs which I am growing this in my house for a long time, and this Ledboria polymorphism in Ledboria rivulata, just a paper species. Okay, now what are those? I will tell you. Now, polymorphism, 4 cytotypes. They have chromosome number right from 30, 45, 60, 90. It is a polyploid series. Of course, polyploids are new species. There is no problem, but this is an autopolyploid. And as you go to higher ploidy, they don't produce seeds. Mostly they are propagating by vegetative means. That means they are not reproductively isolated, or they are not sexual species. They have autopolyploidy. Then Ledboria rivulata has adapted a means of vegetative propagation by formation of bulbils at the tip. Geographical distribution in experimental gardens, study of cytotypes, means of reproduction in Ledboria rivulata supports merger of viridis, karnatakensis, hyderabadenssis under a highly polymorphic species, Ledboria rivulata. Now this, you see me, this is the populations. Now all populations are in front of you. Now each one I can describe as a different species. Am I right? Each one, you see the variegation patterns, flower color, leaf size, leaf shape, leaf venation, each one has a distinct species. Do you agree with this? Will you agree with this? This is all populations, this vegetatively propagating. Answer is definitely a new worker definitely will feel this is a new species. He will describe as a leaves ovate, not linear. Okay. Another will describe leaves produce bulbils. It is a new species. Another will, it is non-variegated leaves. So it is a new species. Is it a right thing for a taxonomist? So I am educating you, those who are young workers, try to understand, see the variations with under cultivation and see whether these differences are maintained. Even these differences are maintained in the right. This is one slide. You see this all variations I am showing, which I grow in my garden. I'm not just talking. I have done it in my house, and then only I am saying it confidently. They don't stand as a distinct. You see, leaves are different, right? Now, what is, see this linear leaf, just like, 2 millimeter, 3 millimeter in a Badami plateau, on Badami plateau. What is it? What these variations are? So it is a highly polymorphic species, which is Ledboria rivulata. Now you see, this is, now I will show you Ledboria rivulata, which has 2n is equal to 30. It is a sexual species which reproduces by seeds. This is a species in our, in this series, and it grows on fields. And you can see this, it is a very common in peninsular India.
Sir, Yadav sir, please unmute your mic, sir.
Is it clear now? Hello?
Yes, sir. Yes, sir. Now it's clear, sir.
Right. Sorry, this will happen. Now you have seen
No
this.
problem. Yes, sir.
See
Sir, your screen is not visible, I guess, sir.
Not visible. Is it visible now?
Yeah, yes, sir. Now it's visible, sir.
Because in
Thank
between
you,
this one is going, so I am sorry. Anyway.
Yes, sir. Thank you, sir.
Oh, what to do?
Hello, sir. There is no one in the lab.
No, I am in my house, sir.
Oh.
So now
Okay.
is it visible now? Right. Sometimes the internet is going, so some little disturbances are there. Now you see this population, Why it is so closely growing? It produces bulbils and it has formed a clump-like structure. See here this from Karst Plateau. Sanjay was telling about Karst Plateau. On this Karst Plateau, if you go, you see these leaves. At the tip of leaf, you find these bulbils. See these bulbils. Very clearly, you can see these bulbils, right? Now see these bulbils which touch to ground and form a new population. What is the chromosome number you see above? It is a 45. Initially, I told you 30. Now it is 45. It is a triploid, and triploid cannot reproduce sexually. That's why it has adapted for vegetative propagation. So this is one I have shown you. Then this Rivulata, see the chromosomes, nice chromosomes, anybody can study. It is one of the very good material for cytological studies to train our students. So 45 chromosomes. Now see this Ledboria with 45 from different locations. It is not one location. Now another population you see. This is from Divaghat. 2n is equal to 60 chromosomes. And see this blotching pattern. And it produces few fruits also. Not fully fertile. 2-3 fruits are developed. So this is a 60 chromosomes. And then you see this is from Badami and other places. So Badami, in Badami population, we get both 45 plus 60 also. Right. Now see this Badami population. Right. See the chromosome numbers in this. Right. Now this is from Yarkar. This population is from Yarkar. See this blotching pattern. Now so much variations in the one species, and see these chromosome numbers. 2n is equal to 45, 60 at Badami. Then this is another population which we collected from Konkan, which has 2n, not Konkan, Junnar, which has 2n is equal to 90 chromosomes. And leaves are about 1.5 feet long. Have you seen? Anybody will be tempted to describe as a new species, which has leaves more than 30 centimeter in length. And that way, all taxonomists in India get confused with bulbous plants. It is a very different group. And we have done 30, 60, 45, 30 different populations have been studied. And then we have come to the conclusion. You can see these chromosomes, different number chromosomes. And that's why when you describe some species, especially in bulbous chromatous monocots, you have to be very careful in delimiting species, okay? Because they have ability to reproduce vegetatively. You can see this all populations, some morphotypes, cytotypes of Ledboria rivulata in peninsular India. So I gave you one example just to illustrate how it is interesting, and therefore studying gives us more idea about species diversity. You can see this meiosis also, which we have done. Karyotypes we have, they are autotetraploid. So that is, Now then, I wanted to correlate how it can be correlated with field. Now the species which grow in Konkan region, this species has 2n is equal to here 90. Okay, here there is a 90, 190. Then when you come to Western Ghats at higher altitude, we have triploid, 2n equals to 45. Then when you come to little drier part, we have peninsular India, we have diploids. And again, when we go to extremely dry region like Badami, again, we have triploids and tetraploids. So this is the way approximately these cytotypes are distributed in different parts. Speciation or evolution of different populations in different parts of our peninsular India. So now this is the species described Ledboria hyderabadiensis. I don't think, because it is having linear leaves, it has no blotching. That is the differences given, which does not stand. And in the same population, we get variations. So they may not stand. So this is about your Ledboria, one genus I have told, in which I have done little work. Now another genus is genus Dipcadi, which is also having tunicated bulbs, and it is another genus, and people keep on describing new species. I don't know. Neither I inspired, even if it comes to me for evaluation, I say this is this species. Even then it is, my worker sees that it is published in some journal, but he wants a new species to his predate without understanding. So there are some new species which have been described in Dipcadi which may not stand the test of time. Now you see this is the key which is given for identification of Dipcadi species. I will not go through this key, but these keys are so, so misleading that no student can identify any Dipcadi species with confidence. Okay, so for example, pedicel stout and pedicel filiform. How student is going to recognize filiform and stout pedicel? Okay, filaments adnate wholly to the periant tube. Filaments adnate in most part to the periant tube. How student can identify? And I have seen dissected flowers. I have seen the filaments are united up to 1/3 part of the perianth, and there is no difference. I have seen all the species. So this key makes, okay, very, misleads identification. These are the species which are described. Now see these chromosomes. I have seen chromosomes to be of great significance in in identification of species of Dipcadi. Why I am telling you? There are 3 series in Indian Dipcadi species. One has 2n is equal to 12. There are about 4 or 5 species which have 2n is equal to 12. Okay. Another group is Dipcadi minor, Dipcadi montanum, which has 2n is equal to 22. And then another group is there, that is Dipcadi, what is that? Dipcadi, this one, Dipcadi serotina, not serotina, Dipcadi, one more, erythrium, which has 2n is equal to 22. So these numbers are of some significance in species identification. You can see distribution, phenology, and chromosome numbers. One series, Govens, Coconens, Serotinum, Saxorum has 2n equal to 12. Ursuli, Montanum, this Montanum, they have 20, and Erythrium 22. Now you know how these species populations remain isolated or reproductively isolated in natural habitats. My observations are very simple, but they are equally important. See, night blooming and day blooming, day and night, blooming time. But the flower smell, if you see, Govens has fragrant flowers, Concanens has fragrant flowers, Serotinum has mild fragrance, whereas Saxorum has very foul smell. Similarly, this one, Montanum, which has very small foul smell. Definitely, there is a stratification of pollinators by the fragrance of flowers, and probably therefore there is no cross-pollination. Pollinators are definite, and these maintain their populations in nature. This is what one small observation. So anyway, these are Dipcadi analysis, and you see this Dipcadi erythrium, which is found in Rajasthan. Now it is also reported from Goa and also from Maharashtra. Probably it has come from Pakistan side, and it has 2n is equal to 22. I have seen all the 3 populations, chromosome number 2n is equal to 22. Then this is a Serotinum, but it is doubtful. Serotinum all over the world, chromosome number shown is 9, means 18. And in our, it is a 12. So probably we need more work, and that will be done in future. So this is from Delhi region. Then most of the Dipcadi species are found in peninsular India, and they are endemic. So here chromosomes. These slides are available. You can see and analyze. So here Cili, tribe Cili, Our high synthesis concentrated in Western Ghats, especially northern Western Ghats. So how many Dipteri species occur in India? So out of 8 Indian species of Dipteri, 4 are endemic to northern Western Ghats, and Dipteri maharashtriensis, which was described, does not stand as a distinct species. Already it is proved also. So we have to be very good, very careful in identification. Now another group, Drimia. In Drimia, I am working for last 30 years. How many species are there in Drimia? Or originally it is Virginia. We call it as a Virginia, which is a very important from, it has cardiotonic glycosides. And you know, this genus, pedicel shorter than bracts, Polyphylla. Is there Polyphylla genus, Dipteri polyphylla in India? Whether anybody confirmed what it is? You know, for last 100 years after Hooke, we are trying to search for Drimia polyphylla. Does it exist? This is the question. You know, when we got the herbarium seed and when we saw the herbarium and flower, 1 or 2 flowers, and when we saw the bracts, it is not Drimia, it is a Dipteri. And unfortunately, it was identified as a Virginia polyphylla. It is a Dipteri species. So nobody could locate this. This is one confusion which we could remove. Then pedicels longer than bracts. This is another category. Pedicels 10 to 35 millimeter, racemose loose. Pedicels 4 to 7, racemose dense. All these are relative terms which does not definitely indicate what is species. Then Drimia polyphylla, Drimia congesta. Somebody described congesta. And now people told Drimia congesta does not occur in India. It is a South African species. So whatever literature has been created in India in last 20-25 years, we cannot believe in identification. And this, you know, that creates a lot of problem for scientists, scientific work. Then Drimia Raji, Drimia Govindapi, Drimia Nagarjuni, Drimia Raujibiki, Drimia Whitey, and so on. So how this is? Now you see, I will explain you. We have published one paper in South African journal on Indian Drimias, and that will clarify your doubts. Now this is one which grows in a coastal region, which has 2n is equal to 30, Drimia indica. Now this is a Drimia Govindapi. From Bangalore it is described. We recognize it as a distinct species. Probably it may be. Then this was identified because congested inflorescence, many of us, including myself, we identified it as a Drimia congesta. And then our, what is his name? Here, I mean, what is his name? Anyway, then the author's name I am not able to remember. This is now described as a Drimia raujibiki. It was a new species and it was described recently. Then Drimia whitey. What is Drimia whitey? Who collected? You know, when after 25 years, I had gone to that, which university, our Bharati Dasan University. And in that campus, for first time, I saw Drimia whitey, which has flowers and leaves at the same time, Sinanthus, and this stands as a distinct species. Then this was Raujibiki, then the Polyantha, then this is I mean this Drimia Nagarjuni is another very distinct species, and that whole was a confusion in our literature. You see, this is Drimia whitey. This is Drimia raujibiki, okay? And we can very easily identify. Now see this same population. Now see this raujibiki, how much variations you get in leaf, shape, size, color, and so on. So this is a little bit difficult. And this species in the Chinar Wildlife Sanctuary, I located from the vehicle only. And I could recognize this is the species which I was in search of. This is Drimia raujibiki. You see the leaves. Leaves are like agave leaves. And it is a very distinct species. And now I came to know it is there in Tamil Nadu and Nagarjuna Hills. See this species. Who will go wrong in identification? Unfortunately, nobody studied them closely, carefully. And therefore, we were, see the stamens, see the flowers, and all that is unique. Okay. So we studied, and now we recognize all these are, I mean, cytological studies which we have done. We need not to go in that. So now we recognize Dreamia coromandeliana, which is a tetraploid, 2n is equal to 40, closely related to indica. It is sexually reproducing, so we recognize. Dreamia Govindappi, okay, from Bangalore. Dreamia indica, Dreamia Nagarjuni, Dreamia polyantha, Dreamia polyphylla does not stand. Okay, it is a wrong. It is a Dreamia. It is a this one Dipcadi. Dreamia raujipiki, Dreamia raji, and Dreamia whitey. Okay, so these are the species which we have recognized, and now this revision has been published. So you can see these chromosomes. There also you get diploids, triploids, tetraploids, and hexaploids. Okay, anyway, so we have seen, if we want to understand that how these species remain reproductively isolated in natural habitats. So one thing you can see, the day blooming species and night blooming species, we can say. And if we cross artificially, some of them cross, some of them do not cross. And that way their genetic relationships can be understood. So we can recognize them as a night blooming and day blooming species. So they are reproductively isolated in nature, and we can recognize them as a distinct species. Cineanthus, okay? So this is which we have done, and that is published already. I will not go in. Now this is one Cili about Cili. Now Aponogeton satarensis. Now this is a species which I described in my live in the beginning, in first time. And see this species which has biforked spike. Then this is the Dipcadi, Aponogeton deckeri, 2n is equal to 56. This is 2n is equal to 26. It is a relict. It is a relict endemic of Aponogeton in India. It has lowest ploidy level. Now I got these materials, from deckeri and satarensis, and I tried to cross them. Okay, this is a female plant. This is male plant, male from satarensis, female from deckeri. It has 56, 26 cross, and I got male plant. That means it is. And then we studied this also, and this way, Madagascar species Aponogeton deckeri is closely related to satarensis. It is proved beyond doubt, and their genetic relationship is proved, and that way it is a example of living witness for continental diphtheria. Pancratium, another genus. I am yet to publish any paper, but a lot of work I have done. You see this Pancratium, and 3-4 species in last 2 years have been published. None of them will stand. Some of you may, those who have described may be listening me also. None of them will stand. What is the problem with this Pancratium species? You see this Pancratium species, the length of corolla tube, the corona, shape of the corona tube, then the stamen filament length, anther length, style included or excluded. All these characters are used for identification of this Pancratium species. They flower in 5-6 days. Just inflorescence starts coming up. In 6-7 days, they complete their flowering and go to sleep. Then you don't find flower. Now, such a group, just by collecting 1-2 specimens and describing, definitely is going to create a lot of problems for taxonomy. Now, I am showing you all this. You can see, and then you decide. Corona, funnel shape, corona, tubular, filaments very long, filaments very short, pollens orange, pollens yellow. These are some differences, definitely. And I am doing this work. You see this size variations in corolla length. Definitely they play important role in stratification of pollinators. Some are night blooming, some are day blooming. Day blooming flowers are without smell. Night blooming flowers are with smell. So there is some very interesting stories, because one genus will take me 2-3 hours to explain. Now you see these coronas, different kinds of coronas. In spite of this, my 20-year study, I have not described anything, because still I am trying to understand what is genus Pancratium. Still I am trying to understand, because unless I have all the materials, I will not say anything. Okay, so see these populations, right? So I have studied all cytology, all of them have 2n is equal to 22, right? Now see these chromosomes. Then meiosis also I have studied. And then, I have tried to see this crossing. All these population except Pancratium, longiflorum, all other species which are recognized in India cross with each other, produce fully fertile hybrids, and these hybrids are also reproducing nicely sexually. Now what to say? From biological point of view or biological definition of species, all these species have to be merged into single species. But as a taxonomist, when they maintain themselves in nature as a distinct entities, we recognize them as a distinct species. So this is a very complex group which I have shown, and this is one example, and you can see this, you can cross any species, and I have about 50 hybrids in my house, and I am studying them, what changes are taking place. Okay, so how difficult it is, right? So one should know. So these are some conclusions which I have drawn. So here, what characters are important, how we can deal with the genus is an important part. Similarly, genus Amorphopalus. Okay, this is the genus Amorphopalus, which has also very interesting stories with chromosomes. And 2n is equal to 33. Now, some of you may be young workers. Do you have diploid chromosome number which is odd? 2n is equal to 39. There are 5 species of Amorphopalus which have 2n is equal to 39. And then why taxonomists recognize them as a distinct species? They are, whether they are triploid or they have odd chromosome number, diploid chromosome number. And all these, which have 2n is equal to 39, do not reproduce sexually. They reproduce asexually. All these ovaries develop asexually, embryos are developed, and they propagate. But we recognize them as a distinct species. Okay. Then, this, you can see these chromosomes. I will not go in all the details because time is over almost. And sometime I have to give it to you and see this, how chromosome different numbers have been evolved in Dipcadis, Amorphopalus species, which we have done. 1428, we have Pionipolis campanulatus, hirsuta. Then aneuploid, 14 minus 1, 13, 13 into 2, 26. We have so many species with 36. Then 26 plus 13, we have 3x, we have 39, Longiconnectivitas, Margaretifer, Bulbifer, Oncophilus, Concanensis, they have 39 chromosomes. So this is approximately evolution. We may not be right also, but just how they must have evolved, this I have shown. So studies in the tropical genus Chlorophytum, same thing. We have a lot of, in this, I am very much impressed that there are 4 species which have 2n is equal to 16 and others with 2n is equal to 14 or multiple of 14. So on one side, we find basic chromosome number 8 in which there are no polyploids. Why this group never showed any polyploid? This is one question. Another, the base number 2n is equal to 7, we have these Chlorophytum members. That Chlorophytum heni, which grows in Kerala, which has 14 chromosomes, and its a close relative is found in Africa, Chlorophytum filipendum, that has 2n is equal to 14. Then we have 28 double Borrelianum chromosome. Then we have 42 Anundanaceum, Belgamensis, Brevicapsum, Glycoides, Glaucum, and so on. Then we have Attenuatum 42, 84, and Khasianum 56, multiple of 14. So why it is happening, how it happened, I am not a cytologist or cytogenetist, but I have tried to understand just from chromosome number. So this is story about chlorophytums, okay? Chlorophytums I have shown, and you can see this evolution of chromosomes I have shown in a, we have shown in a different way how it must have happened. So is it not an interesting story to understand, right? So this is one thing can do. Then Camptorrhiza and Iphigenia. This is a Camptorrhiza is a distinct genus recognized by Q. Still it is not merged by Q. So we described Camptorrhiza indica. Then we described, we have Iphigenias. And you know, this is Camptorrhiza strumorsa, which has a undivided style. And you see this, our Indian Iphigenias have trifid style, All the species are here. This is only Camptorrhiza. We tried to cross them with each other. They crossed, they showed intermediates with all the species. And that way we have shown that it does not stand as a distinct species. And now it has already been merged into Iphigenia species. Okay, even molecular studies support. So anyway, so this is a study which has been done, and only, Glypoclova, similar studies have been done. You can see these two groups in Glypoclova, grass. We have base number 2n is equal to 7 and base number 2n is equal to 6. Now these have different seeds and they are found only in Konkan plains. While X is equal to 7, these species are found on the top of Western Ghats as well as few are in Konkan plains. So there is a definite correlation in ecological conditions and evolution of species and ploidy level. So I will not go. Now you see, on one side we have only single on, which has base number X is equal to 6. Others have two ons, which have X is equal to 7, and intermediate is this one, our, what is this? Glypoclova acuminata. A lot of things can be told, but big things I am showing, which you can understand very easily, right? So those X is equal to 7, they are found on top, and with the X is equal to 6, mainly they are found in Konkan plains. So this way, it indicates distribution also, cytology helps. So this is the evolution we have shown. So anyway, last part, cytocrinum we are working, and you know, all crinums again, these are the crinum species with us. They are very important for galanthamine contents. And see, this is a critically endangered species, Crinum brachinema. Then these are the crinum species, and we tried, this is a new species which was described from Kerala. And you see this Crinum woodroy, which has 2n equal to 22, 44. And no, we have done crosses. Again, all these crinum species cross with each other and produce hybrids. You can see these seeds, right? So this we have done. And these are the hybrid seeds, Crinum brachinema and Crinum woodroy. Woodroy is a tetraploid, this is a diploid. Still they produce hybrids. And this hybridization has been done. And we tried to understand what is a close relationship, what species are closely related, what are distantly related. So this way we have, I showed you examples. Purpose of my lecture is to tell you things are not that easy the way we see. People, you know, with we many times some scientists or our taxonomists say that there is a new species phobia, describing new species without going into details. Just I want some new species on my name. Actually, really speaking, we should try to understand nature, evolution, microevolution, and for that field work, ecology, experimental work, cytology. These are important things. Even your close observations of pollinators, scent of the flower, color of the flower, can they give you how reproductively species are isolated in nature and how they are evolving. I hope I could, because I have gone hurriedly, it takes a lot of time to explain everything, but main things you must have understood. And therefore, to all my young workers and botanists, my request is be critical, don't be superficial, and try to understand. And biology is not that simple. 2 + 2 is equal to 4. It is always different. And that's why biology is one of the very important subject of great potential for research and understanding of nature. Thank you very much. Thank you very much. So if you have some questions, please.
Thank you, sir, for such an informative lecture on biosystematics and its various important aspects. Now, the session is open for discussion. Participants, if you have any queries, you can clarify it now.
Sir, good afternoon, sir. This is Gajalakshmi. I'm assistant professor in Department of Botany, Krishnamurti College. Sir, I have one doubt, sir. Is there any correlation between altitude and ploidy level, sir?
In some cases, there is a definite correlation between ploidy level and altitude. So usually polyploids are adapted to adverse conditions. So at higher altitude, you get mainly polyploids. At lower altitude, where conditions are more favorable for growth, we get diploids. Usually, this is what I am saying. It will not be a rule, but it is a general statement.
So what will be the reason for that, sir?
See, additional chromosome set gives that species more capacity to survive in adverse condition. Additional set of chromosomes or genes. That is what explanation is given. So this is the additional genetic material available with the genome of that species, and that gives them adaptive value, and that way they survive in a somewhat adverse condition.
Okay, sir. Thank you, sir.
Participants, any other queries?
Sir, a participant have asked a question in YouTube Live, sir. How do you see within the species diversity conservation in future anthropogenic and climate change scenarios?
Once again, you please repeat.
Yes, sir. How do you see within the species diversity conservation in future anthropogenic and climate change scenarios?
See, it is a conservation, species diversity and conservation is a in climate change. With climate change, what will happen? Neither I know or most of us, we don't know what will happen to the species. But what I feel, again, it is said that in every species, polyploidy will play major role in survival of those species as a polyploids or as a new species. So major role will be played by through polyploidization of the genome, so that it will be adapting to climatic change, whatever changes come. So that is what it is said also. And of course, it is not my statement, but it is said that polyploidy will play a major role, and that will make those species to survive. However, this climate change is happening in a very short period of time. Whether these species will get opportunity to polyploidize and survive, that only God alone knows, or nature alone knows. Yes?
Sir, excuse me, sir. Sir, I'm Jalil from Trichy, sir. Thank you for your wonderful insight, sir. Here I have one doubt, sir. Nowadays, the taxonomy curriculum, which is in the level of alpha taxonomy level. But when we construct the curriculum of taxonomy in the practical level, but how we should construct it in omega level, omega taxonomy, that is by experimental level.
That, see what I feel, that we have wonderful materials in our country. Suppose in your college, if you collect just chlorophytum species and keep them in a pots, small pots, okay? And if you ask them to hybridize them, study their chromosomes, and try to correlate, definitely they will understand polyploidy. They will understand whether species hybridize, interspecific hybrids take place. If these hybrids, if you produce, grow them in next year, ask students to understand what changes have taken place. So there are wonderful materials. Only thing that we are exam oriented and mark oriented. That's why whatever is done in universities and colleges, I have seen, it is just to make them to get degrees, not to understand subject. I am a teacher for last 30, 35 years. I have seen this tragedy because they want only how to get good marks and how to get degree, not to understand. So education, I'm sorry, you are also teacher. I am also teacher, but now education has become something different. It is not for learning. Usually it is for only thought that education is for earning. So I learn to earn only, but not knowledge as such. So under this condition, it is a teacher who can make difference. Teacher can make difference and make student to understand and go on right path.
Yes, sir. Thank you, sir.
I will just add a few things to that, Dr. Yadav.
Yes,
I have never taught in any university or anything, but to create facilities for omega taxonomy is a tough task because basically teachers themselves do not have the idea what omega taxonomy is and what are its requirements. And none of the universities have even basic facility for, alpha taxonomy.
Right. Yes, sir. Yeah.
To create infrastructure for, omega taxonomy is a tough job. It depends on a teacher who is really trained in omega taxonomy and then undertakes. For example, Dr. Yadav's lab, he has created facilities and the students follow and undertake research further in this area. So one has to visit such labs and see what facilities they have created to teach omega taxonomy or molecular taxonomy or any advanced taxonomic levels. But in our country, the basic taxonomy or the alpha taxonomy is not taught properly, in fact. So that is, if we graduate at alpha taxonomy, then only you can go for omega taxonomy. Usually, there are facilities for research scholars to undertake omega taxonomy, and some teachers who are interested, they suggest such problems and they undertake. So some of the teachers should create facilities. Am I clear?
Yes, sir.
Any other question?
Participants, if you have any other question, you can ask.
Akshaya, there is no more questions, Akshaya.
There are no questions. Good. That means I don't know whether whatever I have said, whether participants have got what I wanted to say, because literally, I have gone very hurriedly. That is true, but no, time is also an important factor. But some idea you must have got, and I hope we should. Because I have seen in India cytological work, almost it is standstill now. Nobody lab. Same like classical taxonomy also. At least there are some people, but cytology, nobody is doing classical all are molecular and, only they want this one, barcodes, molecular markers. But, those do not give you any idea exactly how species work in nature. Only that can give you relationship, but it will not give how evolution is taking place in nature. And therefore, these biosystematic kinds of studies only can make you perfect in understanding natural evolution, not molecular biology. This, you take it from me. Even if you give me entire genome, I will not understand how that species looks like, how it behaves in nature, how pollination takes place. It will not be represented in molecular biology. And therefore, we should not leave roots of our subject and just hang out the branches. That is my message to you. Thank you very much. If there are no questions, then we will disperse.
Yes, sir. Thank you, sir, for clarifying all the doubts. Now I request all the participants to take a lunch break and rejoin by 2 p.m. for the next technical session.
Good afternoon, sir.
Good afternoon, madam.
Sarah, shall we start? Sir, sir, your mic is muted, sir.
Yes, madam, I am ready.
Yes.
If
Yes,
participants
sir.
are ready, I am ready.
Okay, sir. Shall we start, sir?
Yes, by all means.
Yes, sir. Thank you so much, sir.
Thank you,
Sarah?
madam.
Yes, ma'am. Yes, ma'am. We'll start.
Good afternoon, and I once again welcome you all for the virtual lecture workshop organized by the Department of Botany, PSJ Krishnamal College for Women. Now I request Dr. K. Gajalakshmi, Assistant Professor, Department of Botany, PSJ Krishnamal College for Women, to welcome the speaker of Technical Section 3, Dr. Mohammad Umar Sharif.
Good afternoon, one and all. With great pride and honor, I am here to introduce to you Dr. M.U. Sharif, the guest of honor and speaker for Technical Session 3. Our guest speaker today, who is simple, very energetic and dynamic, was a nature of Mysore. He obtained his graduate degree in Science from Yuvraj College in 1984. His master's degree is from University of Mysore in 1986 with a specialization in cytogenetics and plant breeding. Later, he pursued his research on aromatic and medicinal plants of Karnataka under the guidance of Professor K.S. Jagadish Chandra. He was awarded the doctoral degree in the year 1994. Later, he served honorary lecturer in Mysore University and lecturer in botany at St. Philomena's College, Mysore. He served as senior field officer for 5 years in Rubber Board, Mangalore, Government of India. By his constant efforts and zeal, he was selected as Senior Scientist in Botanical Survey of India, Government of India, Calcutta in the year 2001. He was transferred to Andaman Nicobar Circle Office at Port Blair. He turned out a very interesting study on the ethnobotany of the aboriginal tribes of the Andaman and Nicobar Island. During this study, he recorded many of the interesting and potential plants used by these tribes in their day-to-day life. And this work was submitted to Government of India, New Delhi. In 2005, Dr. Sharif took the charge as head of National Orchidarium and Experimental Garden in Erkard. In 2008, he was promoted as Scientist D by the Ministry of Environment and Forest in New Delhi. In 2013, he has been given the charge as Deputy Director of AJC Bose Indian Botanical Garden, Calcutta, and carrying out work on survey, introduction and conservation of red plants with emphasis on orchid flora of Northeast India. In 2015, he has been promoted as the Joint Director and holding the charge as the Curator of AJC Bose Indian Botanical Garden, Howrah. Dr. Sharif has attended several conferences and seminars of various national and international repute and presented research papers. He published 45 research papers in tissue culture of aromatic and medicinal plants, floristic diversity and ethnobotanical studies of Andaman Nicobar Island, and orchid diversity of Western Ghats. He has co-authored in a book, Jarvas of Andaman Island, which is published by BSI Calcutta. He has edited 7 angiospermic families for the second volume of Flora of Andaman and Nicobar Island, which is preparation at BSI Calcutta. He also the team leader for the forthcoming volume 28 of Flora of India, which involves 24 angiospermic families and about 504 taxa. In 2017, he promoted as head of the AJC Bose Indian Botanic Garden, Calcutta till 2019. From 17 September 2019, he has joined the Botanical Survey of India, Southern Regional Center, Coimbatore, as Joint Director and Head and serving the Coimbatore office. Presently, he is working on the project Flora of Kerala. With this amazing credibility and expertise, Dr. Sharif enlightening you and delivering the lecture undoubtedly. I welcome you, sir, for taking this technical session 3. Thank you all.
Thank you, ma'am. Now I call upon Dr. Mohammed Umar Sharif, Scientist E, Head, Southern Regional Center, Botanical Survey of India, to deliver his lecture on the topic Conservation of Rare and Endemic Orchids of Western Ghats.
Sir, please unmute your mic, sir.
Thank you very much. Thank you very much, madam. And I am audible to all.
Yes, sir, you're audible, sir.
Yes, sir.
Okay. At the outset, I thank the convener of this academic virtual lecture workshop, Dr. M. Sanjappa, and ex-director of BSI in general. And in particular, I thank the authorities of Krishnamal College for Women, Coimbatore, which is a college of excellence. And especially, I thank the principal, Dr. Nirmala, and the head of the Office of Botany, Dr. Krishnaveni, and the coordinator of this today's virtual lecture series, Tamil Selvi, for giving me an opportunity to speak and to share my experiences, what I have just gained with regard to orchids when I was in work in Botanical Survey of India. In addition, I also thank again the Krishnamal College authorities for giving me an opportunity to share my thoughts and to speak about the taxonomic topic amongst the well-known, renowned taxonomists like Dr. M. Sanjappa, Professor S.R. Yadav, and Maimel Sabu, and Dr. Sampath Kumar. Thank you very much, and I thank all the participants for just have a patient hearing for this today's lecture. With this, I will just share my PowerPoint. Give me 2 minutes for sharing my PowerPoint. Okay. Is my PowerPoint is clear to all?
Yes, sir. It's clear, sir.
Yes, thank you very much. So, friends, today I will be just speaking on the topic conservation of rare and endemic orchids of Western Ghats. A very fascinating family and a very well-developed family, Orchidaceae, in which these plant jewels or the botanical jewels are just included. So since I am coming from Botanical Survey of India, a just brief introduction of Botanical Survey of India, especially for the students who are participating in today's this webinar lecture. Botanical Survey of India, coming under Ministry of Environment, Forest and Climate Change, is an organization committed to document and conserve the floristic diversity of our country. The very important component of any nation under consideration, a sovereign property, a sovereign property of any nation under consideration, and a very, very important component of essence of life on this Mother Earth. This important component is to be protected, and the prime responsibility is lying with Botanical Survey of India. This Botanical Survey of India is having its headquarters at Calcutta. Apart from having a headquarters at Calcutta, it is having 4 divisions at Calcutta. That is, one is A.J.C. Bose Indian Botanic Garden, under which again one Central National Herbarium, and then we are having Central Botanical Laboratory and industrial section of, and we are having one industrial section of Indian Museum in Calcutta. 4 divisions apart from headquarters are existing in Calcutta. Besides this is having 11 regional circles distributed in different parts of our country, like Coimbatore, which is Southern Regional Center, Western Regional Center at Pune. Then 5 years back, we have got Hyderabad's Deccan Regional Center, then Central Circle at Allahabad, then the East Zone Circle at Jodhpur, Northern Circle at Dehradun, then we are having Gangtok Circle at Gangtok, then Shillong Circle we are having, Arunachal Pradesh Circle at Itanagar, Andaman Nicobar Regional Center at Port Blair, and recently in the last year, November, new circle or new regional center has come up at Solan, Himachal. So these are the 11 regional centers who are working on the conservation of the important component, the floristic diversity, or in fact, in general, biodiversity. This Botanical Survey of India is having 5 important objectives, and these objectives are collection, identification, and conservation of floristic diversity of a country. The second objective is publication of regional floras. Whatever the knowledge that is gained with the survey and explorations that is carried out in the forests, this is published in the form of regional floras. Then the third objective is setting up of regional herbarias and museums. Then the fourth objective is to conserve and multiply rare and endangered plants. And the fifth objective is documentation of traditional knowledge or tribal knowledge. So today's lecture falls in the first objective, collection, identification, and conservation of floristic diversity of country. So this important component, floristic diversity, this important component, floristic diversity, is conserved by two important aspects, that is two important ways. That is, one is in situ conservation and the second one is ex situ conservation. In situ conservation is conservation of plant entities in its original habitat, either by reserve forest, by biosphere reserves and protected areas it is conserved. And then coming to second part, ex situ conservation, lifting the plant entity from its original habitat and conserving it at other places called ex situ conservation. Again, in situ conservation is carried out by three important methods. One is by setting up of biosphere reserves and protected areas. Totally in India, we are having 18 biosphere reserves and 50 tiger reserves are existing. And apart from this, if we go for national parks, we are having 103 national parks. And by setting up sanctuaries, 543 sanctuaries, the plants are being conserved by in situ means. Coming to ex situ conservation, it is conserved by again three methods. One is by botanical gardens, the second one is by tissue culture, and the third one is by gene banks. The botanical gardens are living repositories of plants. Let us not go deep into that one. Of course, I'll be talking about experimental garden, national architecture and experimental garden. At that time, you come to know about how important is these botanical gardens for conservation of plant entities. The second part is by tissue culture. For that one also, I'll touch upon a little bit during my lecture. And the third one is by gene banks. Of course, the biggest gene banks in India is from ICRISAT, Hyderabad, which conserves the agrobiodiversity of many crop plants. So this is about the methods of in situ and ex situ conservation. So with this background, let us go further and now come to the National Archidium Experimental Garden, which I was holding the chart from 2005 to 2013. This much period I was just holding the chart of National Archidium. At that time, I happened to carry out some biosystematic work on orchids of Western Ghats. So this National Archidium and Experimental Garden is initiated in the year 1963 with an area of 25 acres in Shivarayan Hills of this Salem district. And this is here we are conserving. We are well known for conservation of orchids of South India, as well as it is also known for the conservation of RET plants of this, especially the peninsular India. This is the view of this National Archidium. I'm just with a famous archaeologist, Dr. C. Satish Kumar from Tropical Botanical, Jawaharlal Nehru Tropical Botanical Garden, Trivandrum, who has visited garden for just observation and suggesting some improvements. So this garden, apart from this, I told, apart from conserving orchids, this garden is well known for conservation of RET plants also. And this garden is having a record of conserving the pitcher plant from last 40 years. The maturest of the mature pitcher plant, Nepenthes khasiana, which is a native of Khasiya and Jaintia Hills, he has been conserved in National Archidium and Experimental Garden haircut from last 40 years. And this is the view of this Nepenthes khasiana, which is in a enclosure. And the cross-section of pitcher plant has been taken to show the trapped insect in the previous night. A cross-section has been taken, and this is how it is conserving. It is just trapping the insects. So here are some of the glimpses of this National Archidium Experimental Garden, some greenhouses, the glass houses, and the pathways which has been laid in National Archidium and Experimental Garden. So with this background, let us come to the today's particular topic, that is orchids, conservation of RET, the conservation of rare and endemic orchids of Western Ghats. Friends, I told Orchidaceae is a very fascinating and well-developed family, having very, very unique plants, which are regarded as the aristocrats as well as the novelties in the plant kingdom. They are the botanical jewels. Orchids are of ornamental importance, and they are of they give the flowers of exquisite beauty and variety to the mankind. Orchids have always been seen as the symbol of love, beauty, grace, nobility, and elegance. The incredible range of diversity in their size, shape, structure, and color of the orchid flowers records their position as botanical jewels in the plant kingdom. They are very fascinated. Their colors, their positions, and their shapes of the flowers are so fascinated, and they record the position as botanical jewels in the plant kingdom. Coming to their economical importance, orchids account for multi-million-dollar floricultural trade world over as cut flowers as well as pot plants, and they nearly share 8% of the international flower market. Along with the incredible shapes and fascinating shapes, the very important aspect of the flowers of orchids is they are having longest vast life. The vast life of the flowers of orchids have further enhanced their value among the horticulturists, garden enthusiasts in the world over. One Dendrobium sony in the National Academic Experimental Garden, we noted the date of the flowering, and then we saw that date of the just shrinking, and then it was nearly for 40 to 45 days that was completely fresh. So that's why the orchids have got the long vast life. That's why they are very much preferred in the field of this cut flowers and horticulture industries. So here are some of the photographs to show the fascination of these orchid flowers. Of course, many of them are exotic plants. Just to show the fascination of this important flower group, I'm just some 4 or 5 slides I'm showing. This is the cymbidiums in the cut flower industry. This is the color and marvelous appearance of this cymbidiums. Then we are having Tapepidaliums, lady sleeper orchids, very unique, and show very exaggerating or very unique coloration and shape of the flower. Then we are having Phalaenopsis, generally called moth orchid, and very unique color and striations are so fascinating, and that's why they are regarded as aristocrats in the plant kingdom. And again another Phalaenopsis. Then we are having Vandas, very beautiful striations and very beautiful spots. We can really have a look of these fascinating colors and shapes of the flowers of these orchids. This is again blue Vanda, and in exotic and hybrids. Of course, hybrids, there are 2 important aspects of these orchids. Again, the wild orchids and the hybrid orchids. So now I'm not touching the hybrid orchids. Of course, that itself is a very big aspect to just understand. Now, just let us, with this background of this coloration and the excitement of these flowers, let us go towards the wild orchids of Western Ghats. Coming to this distribution of these orchids, geographically orchids are distributed worldwide with greater concentration in tropical and subtropical regions of high humidity. They are highly adapted plants and distributed in temperate and tropical countries except deserts and these polar regions. They are found distributed in almost all parts of the world. Mexico, Central and South America and Asia are rich in orchid population. About the rainfall and about the temperature regimes, they prefer to have a rainfall of about 1,500 to 2,500 millimeter per year, distributed over long period from April to October, and relative humidity should be required of 70 to 95%, and temperature range from 10 to 15 degree provides optimum climatic conditions for the majority of the orchids. This is the general tendency of these orchids for survivability. Now, where do these orchids occur? Orchids are, I told, they are cosmopolitan. They are very well adopted plants and distributed in almost all parts of the world. That's why they are cosmopolitan, occurring in almost every habitat apart from deserts, marine and glaciers, that is in polar regions. The great majority are to be found in the tropics, mostly Asia, South America and Central America, but they are also found above the Arctic circles in southern Patagonia and even on Macquarie Island close to Antarctica. They are very close to polars but not found in polar areas. Then coming to India, we are having the hotspots for orchids, and these hotspots are, one is Western Ghats, then this is the Western Ghats, then we are having Western Himalayas and Northeastern Himalayas as well as Andaman and Nicobar Islands. These are the 3 hotspots with respect to the orchid population. So these are the 3 hotspots of India which has been shown here. Another important aspect of these orchids, apart from the beauty of these orchid flowers, these orchids are well known to show very important aspect called biomimicry in animation, different animation forms. The most intriguing feature of the orchid flowers is their striking resemblances to various forms of animal life to attract different pollinators to bring about deceitful pollination. They mimic the shape, pigmentation, and even exhibit particular scent given by the female insect. The male is in search of a mate, is attracted by the flower and not only cheated by its pleasure, but is made to fly away with a load of pollen grains. So to carry out deceitful pollination, different shapes of the flowers has been attained in this family Orchidaceae, and based on the shapes of these bees in the form of donkey, in the form of monkey, in the form of dog, in the form of lizard, in the form of eagle, in the form of bee, they have been nicknamed accordingly, like dancing girl orchid, which belongs to the genus Oncidium. And then we are having this bee orchid in the form of Ophoris epifera, then donkey orchid Diuris longifolia, the flower of the shape gives a resemblance of donkey. And then we are having monkey orchid in the form of Orchis simia. Then we are having spider orchid belonging to the genus Arachnis. And then we are having lizard orchid in the form of Himanthoglosum hircinum. And then like this, continuing this biomimicry aspects, on the petals and sepals of this orchid, it is referred to as Dracula orchid, belonging to the genus Dracula, species Chestertoni. And then apart from continuation from this one, we are having further a moth orchid in the form of Epigenium amplum, a green spider orchid in the form of Hebenaria multica, in the form of the Hebenaria ovalifolia. Then we are having pigeon orchid in the form of Peristeria alata. Inside the petals, there will be the pigeon. Just since this is a fully white flower, little this one focusing is little problem in this Peristeria alata. Then we are having a very important jewel orchid and so endemic to South India, Anectical celatier, and it is referred to as white ant or the termite orchid. Then we are having butterfly orchid in the form of Papilantha terris, and even another butterfly orchid also is there, I will be showing. And we are having laughing orchid in the form of Phalaenopsis parishi. Then we are having bull's head orchid in the form of Silogyne ovalis, and then we are having sparrow orchid in the form of this area Andamanica. Then here is the parrot orchid in the form of Bulbophyllum rocheldianum. To continue further, we are having lady sleeper. I told very fascinating shapes they attain. They attain lady sleeper orchid in the form of Papapedalium spicerianum, Papillum insigni, Papillum villosum, Papillum hirsutissimum, Papillum fairianum, and the only South Indian orchid of this lady sleeper category is Papillum drury, which is having black band in the on the petals and sepals. Then we are having duck orchid in the form of Thynia bicornis. I told the dancing girl orchid in the form of Oncidium goldianum. Then we are having eagle orchid, as though the eagle lifting the flower, the chick from its beak. This is called Stenophia wardii. So these are the different shapes which has been biomimicked by these orchid flowers to carry out deceptive pollination, to attract some of the pollinators, to bring about the passage of this pollinia, the transfer of pollinia from one flower to another flower. So they again the biomimicry continued. Cricket orchid in the form Brassia cordata, butterfly orchid again Psychopsis papilio. The appearance of the butterfly orchid is like this. Then duck orchid again, one more duck orchid, Cliena major, Cliena major is the duck orchid. Then deer's head orchid Calypso bulbosa. Then dog orchid in the form of Podonorchis lesonii, and then fire orchid Renanthera inshutiana, which is a rarity species found in northeastern India. Renanthera inshutiana, also known as red vanda. So there again, still little bit continuation. Foxtail orchid in the form of Renanthera inshutiana, the foxtail orchid, a very important ornamental orchid used by some of these ladies in the northeastern area for ornamenting their hair. Then we are having mosquito orchid in the form of Habenaria andamanensis. Then we are having this mouse tail orchid in the form of Oberonia brunoniana, again endemic to South India. And we are having a very small and a minute orchid which forms just by the assimilatory roots, or and also called a snake orchid, called Tinophyllum alvizii. And then we are having this bumblebee orchid in the form of Catonia bicornis, and then again monkey orchid in the form of Orchis simia. So besides having the ornamental importance, these orchids also are well known from the long ages to possess very important medicinal plants. Charak, in his book Charak Samhita, very long years ago itself, has given the importance of orchids with regard to medicinal properties to cure many of the ailments with respect to herbal remedies. So just to know the importance, besides the aesthetic importance, they also possess the medicinal importance. And some of the important orchids has been highlighted in the form of Acampia primosa, is used for the herbal as a herbal drug in rheumatism. Then it is used in Cymbidium alveolum as emetic, to especially lead for the vomiting, especially in the cases of this food poisoning or just poisoning cases. It is Cymbidium alveolum is referred for as emetic. Then we are having Cymbidium elegans in nervous disorders. Dendrobium nobai, the state flower of Gangtok, is used in eye troubles and curing the wounds. Then Erium musicula in heart problems, it is referred to as in some of the herbal remedies. and then Eulophia nuda as a blood purifier, Geodorum densifluum, a terrestrial orchid of very commonly seen in Western Ghats. As soon as the rain stops, immediately Geodorum densifluum comes up and it is used as insecticides. Hebneria acuminata used as tonic. Liparis odorata used in burns and ulcers. The Incostalis reducer, the foxtail orchid, is used in rheumatism and emollient, especially for smoothing the skins. And then Cetarium nepalens in the curing of the malaria, it has been referred to as. Then Vanda cerula, the blue orchid of this northeastern India, is used in eye diseases to control, to rectify cataracts and glaucoma. Then we are having Vanda testacea, again a common orchid found in Western Ghats, is used in the herbal remedies of rheumatism and nervous disorders. So further, if you continue these orchids have a very poor fossil history, and they have originated in Southeast Asia some 40 million years ago. Theophrastus is the first to refer a group of plants called orchis in his book Enquiry into Plants. The name orchid is derived from the Greek word orchis, meaning testis, and refers to the parent tubers of terrestrial orchid genus Orchis, as well as in some of the Hebeneria species and other similar genera. Orchids constitute one of the highly evolved, diverse, and successful group of flowering plants. Orchids show extremely incredible range of specialization in floral parts, which are unparalleled in any other groups. Their flowers, even though they look very, very complicated, they are very simple. They are having the segments distributed in two parts, and in two parts. The first segment is referred to as sepals, which are outer sepal and the middle sepal, and these are three sepals will be there. And apart from that, the inner sepal, in the inner whorl, we are having these petals. Out of the three petals, one petal is modified into this lip or labellum that I will be showing in further slides. Coming to the history of this Indian archaeology, after knowing this Indian archaeology, we'll go to the important the structure of this flower, and further we'll continue. So now coming to the brief history of this Indian archaeology, Indian orchids were brought to the notice of the world by Charak, the great Indian medicine man, as back as 125 AD, who described Vanda and several other orchids in his book Charak Samhita. Then the first scientific account of Indian orchids was given in Hortus Malabaricus by Van Reed, and then the Dutch governor of Malabar, and he described 8 species in this Hortus Malabaricus. After that, the contribution came from Dr. William Rasborough, the father of Indian botany, and who is the first salaried superintendent of A.G. Bose Indian Botanic Garden. He made a set of 2,595 life-size color drawings of plants for his Flora Indica, and provided a treatment of 57 species of orchids in his book Flora Indica, Volume 3. If you continue the Indian archaeology, Dr. Nathaniel Walich also has contributed in the form of Tentamen Florae Nepalensis Illustrae, Planta Asiatica Rariots, described several orchids and provided beautiful illustrations by the artists like Vishnu Prasad, Gorachan, Lakshman Singh, C.M. Curtis, Miss Derek, R.K. Grivelle, and John Lindley. Next, if you further continue, 1828, a numerical list of dried specimens, popularly known as Walish Catalogue, in which more than 20,000 specimens belonging to subtribe Gudrine in the catalogue were either collected by Walish himself or from other collectors, including collections by Roxburgh, Gomez, Griffith, White, etc. in this Walish catalogue. If you continue further, in 1799 to 1858, illustration on the botany and other branches of natural history of the Himalayan mountains was done by Royle, and he credited Professor Lindley for several new texts of the orchids. Here is the John Lindley's photograph I have just. Then further to continue, we are having this contribution to the archaeology in India, number 1, in which 126 species of orchids has been described. Further, 1810 to 1846, William Griffith, in his book Notoliae ad Plantas Asiaticas, and then itinerary notes of plants collected in Khasi Hills and Bhutan mountains in 1848, and Icons Plantarum Asiaticum are some of the finest works on Indian, including orchids. Griffith provided accounts of 147 species of orchids under 49 genera, and recorded mostly from Khasi Hills and its surrounding areas. Further, if you continue, in 1817 to 1911, Sir J.D. Hooker has contributed a lot with regard to this flora of British India. He has made descriptions of thousands of species of orchids, and then in Hooker's Icons Plantarum, volume 22, also he has described many of these orchids of India. A Century of Indian Orchids by him is also a well-known record with regard to this Indian archaeology. Impressed by the work, Sir George King and Robert Pantling also they contributed for the orchids of Sikkim Himalayas in the year 1840 to 1909, and this is in which 448 species of orchids has been described. Further continuation of the work, we can quote the work of John Sriminger Dhuti, Professor Dhuti in 1845 to 1922 brought out the descriptions of some new species of Orchidaceae from northwest and central India. And further, in which 12 species has been described. The orchids of northwestern Himalaya with 173 species, also a very good record of Professor Dhuti. And then if we continue further and coming to our contribution of Botanical Survey of India scientists, further Santapo is and Kapadia, they have brought out the book The Orchids of Bombay in the year 1966, and Dr. A.S. Rao, Orchids of Khasi and Jayanta Hills in 1971, and Orchids of India in the year 1979. They have contributed for the Indian archaeology. Then came the contribution from Dr. J. Joseph, who was just a scientist in the BSI SRC in 1928 to 2000. After doing the this one plastic studies in Nilgiri and the Western Ghat area, he brought out Orchids of Nilgiris, a small book but very useful book with regard to identification of these orchids. In 1987, he published the Orchids of Nilgiris. Further to add to these contributions of archaeology, Dr. S.N. Hegde, who is from Bangalore, he has contributed in the form of Orchids of Arunachal Pradesh and Orchids Conservation, Culture, Farming and Trade in 2001. He has released this account of these orchids. Further, Professor Mr. U.C. Pradhan in 1949, he brought out Indian Orchids Guide to Identification and Culture in volume 1 and 2 in 1976. And Dr. A. Nageshwar Rao, he has contributed a lot in the North India, and he has quite instrumental in setting up an orchidarium in Chessi of this Arunachal Pradesh. And his PhD thesis itself was on studies in the family Orchidaceae of Arunachal Pradesh. Then Dr. S.K. Katki, Orchids of Meghalaya, a very good compilation of this work by Katki, and he has brought out a book on ladies sleeper orchids of India in 1984. Continuing further, Sandhya Jyoti, the ex-Joint Director of Botanical Survey of India, she has contributed a book on Orchidaceae, especially with regard to genus Coelogin. And then we are having the contribution of N.P. Balakrishnan and Dr. R. Ansari, who are from BSI, and they have contributed for the genus Oberonia and published in Orchid Monographs, Volume 4. Dr. N.P. Balakrishnan also is further, his work is also known very well with regard to the orchids of Andaman Nicobar Islands. Also, he has compiled much of the work of this orchids of Andaman Nicobar Islands. To continue, I.C. Barua for Orchid Flora of Kamrup district in 2001. Somdev and Naithani, they have brought out in 1986 the Orchid Flora of Northwestern Himalaya. Sharat Mishra, an engineer from Orissa, and out of his own interest, he has just expertise in orchids, and he has brought out the books of Orchids of Orissa in 2004 and Orchids of India in 2007. Instrumental in reporting many of the new species from Orissa also. H.J. Chaudhary, the ex-Joint Director of Botanical Survey of India, he has brought out Orchid Flora of Arunachal Pradesh in 1997 and a century of West Himalayan orchids in 2013. And Satish Kumar, a well-known friend of Dr. Sanjay Pajala, he is also quite instrumental in bringing out the catalogs of Indian orchids, a student of Professor Professor Manilal. He has brought out work on conservation assessment, catalog of Indian orchid in 1994 with the conservation assessment and management plan, and he has brought out this conservation assessment and management plan work report for endemic orchids of Western Ghats, and he has contributed for orchids of Kerala in 2004 and an orchid digest of Manipur, Northeastern India in 2005. So with this history of this Indian archaeology, we'll just go further in the study of this Orchidaceae. If you see the taxonomy of these orchids are monocotyledonous plants and they belong to family Orchidaceae, which includes 820 genera with 25,000 species distributed all over the world. Orchids constitute, I told, 8% of the flowering species and 60% of monocots. In India, about 1,200 species in 178 genera are recorded, and number is keep on changing because of new additions and deletions. Further, in Western Ghats, if you take into consideration, we are having 250 or 245 species belonging to 30 genera has been reported. And apart from this wild orchids, the progress in the hybrid orchids is so fast, and more than 1.1 to 1.5 lakh registered hybrids the cultivars exist presently in the flower market. I told these orchids are very unique with regard to their morphology, with regard to their anatomy, with regard to their growth pattern, habitat, all are quite unique. If you take anatomy also, it is unique. If you take anatomy, the association of mycorrhizal fungus is quite interesting, and their phenology and the flora-biology also quite unique. Once if we take the habit, they generally show epiphytic. They show epiphytic habitat. So of course, some are terrestrial species and lithophytes are also existing, but commonly they show epiphytic habitat, and they are having very unique pattern of growth in the form of monopodial growth and pattern, and they show sympodial growth pattern. Especially the many of the terrestrial orchids, they show monopodial growth, whereas the epiphytic orchids they show sympodial growth. In monopodial growth, we extend the shoot these plants to extend the shoot tip and grow, producing new leaves at the tip, and the main axis grows. Whereas in sympodial orchid, grow laterally where new shoots are produced from the base of the previous year, and they just horizontally they grow and they just propagate themselves. And apart from that, they show very uniqueness in their habit also. They produce very specialized structures called pseudobulbs. They produce some of the very specialized roots in the form of velamen or the aerial roots, which will have this velamen tissue which absorbs moisture from the atmosphere and give to the plant for helping its growth. So they show the presence of many sympodial orchids produce peculiar shapes or clumps called pseudobulbs, which serve as storage organs for food and nutrients. Pseudobulbs may be of different shapes. They may be cylindrical, spindle-shaped, egg-shaped, spherical, barrel-shaped, and button-shaped. And apart from that, the size of the pseudobulbs also varies, very minute of 1 to 2 millimeter to 3 to 4 meters, especially in the case of this vanilla species, Vanilla walkeri, the pseudobulbs are 3 to 4 meters in their length. These pseudobulbs may be as small as 1 to 2 millimeter, as in Bulbophyllum minusissimum and then Oberonia affinis, and then we are having this Tenophyllum albizae, which are very difficult to locate in the forests. Apart from that, we are having very long of 30 meters to 10 to 20 to 30 meters in Vanilla walkeri, and then we are having a very robust orchid in the form of Grammatophyllum speciosum that I will be showing some of the photographs in my future slides. So coming to habitat, orchids exhibit terrestrial, epiphytic, lithophytic, and saprophytic habits. And these terrestrial orchids are rooted in soil. Example, Anaptochellus elatia and then Hebineria, they are rooted and they are just grow and show their monopodial growth of their pattern. Then coming to epiphytic orchids, they cling to the support like trees. Most of the epiphytic orchids produce aerial roots in the form of filamen. And then examples are Dendrobium, Cymbidium, Bulbophyllum, whatnot, Diplocentrum. We are having Cymbidiums, we are having Coelogenes. They all epiphytic orchids, they produce this aerial roots and cleave to the support and they grow. Coming to lithophytic orchids, which grows on orchid with meager layers of soil or the humus. Some of the species are Vanda, Coelogene, Anthogonium. They just grow on the rocky surface. Even we have located Bulbophyllum fuscopurpureum, an endemic orchid of Western Ghats in some of these tea estates of Munnar. Big rocks are fully covered with lorry loads of this Bulbophyllum fuscopurpureum and Bulbophyllum fisheri and Bulbophyllum tremulum, all those things. So apart from that, they show saprophytic orchids. Some of the orchids are saprophytic also in their growth, and they grow on dead and decaying organic substances. Some examples are Eulophia and Epipogium. These are some of the saprophytic orchids also. They exist in this family Orchidaceae. Coming to orchid flower, I told they are, even though they look very complicated, they are very simple. They will have 3 holes. Orchid flower contains 3 sepals. Here is the sepal, 1, 2, 3 sepals. Outer hole is form sepals, and the inner hole of 3 segments form the petals. 2 petals are there, and 1 petal is modified into a specialized structure called lip or labellum. This is a very important feature of this orchid flower. And here the ovary is inferior, and apart from that, there are 2 important modifications in this flower, and that is the presence of gynostemium. The style and the pistil and style, they fuse together. The stamens and the style, they fuse together to form an important structure called gynostemium that I will be showing in future slide. The lip or labellum, I told, 1 petal is modified into a very important specialized structure called lip or labellum. Lip or labellum is one of the main elements that makes orchid flower recognizable as such. It is very much helpful in recognizing some of the orchid species. They are movable or firmly attached to the column foot by their base. They may be sessile or with a claw, simple or divided into various lobes, and are mostly decorated with some tissue deposition known as callus. These are some of the tissue depositions will be there. Keels are lamellae, or sometimes with different types of hairy outgrowths. The basal, middle, and distal portion here, the basal, middle, and distal portion, which are referred to as hyposhell, mesoshell, and epishell respectively, is also present. Lips show very unique type of color combination, completely different from the other perianth segments. It is a very attractive part in the orchid flower, and very fascinated. Based on that, we can take some clues for identifying the flowers of this orchid plants. The margin of lips vary from entire clayate and serrate to crenate, dentate, and fimbriate type. And then I told the another modification in the leaf is the male and female sex organs are fused to form a compact structure called column or gynostemium. This is the structure of this gynostemium. Even here, the column or gynostemium is the combined structure of style and staminal filaments united together, and is the most obvious structure in family Orchidaceae. Degree of union increases from primitive Apostosoides and Crepidioides to the advanced Epidendroid and Vandoid groups. Column shows remarkable range of variation in its posture, shape, and size related with the anther and stigma it bears. And then another important character of this flower of this orchids is presence of pollinia or the pollinarium. The pollen grains are present in a sac-like structure or bag-like structure called pollinia, and a complete structure is referred to as pollinarium. The compound structure formed by the pollinia, caudicle, stripe, and visidium is known as the pollen apparatus for pollinarium. The presence of pollinarium also an adaptation of the flower for just helping in pollination by the pollinators visiting the orchid flower. The pollen grains in orchids adhere with each other by means of certain viscid or waxy substances and form a definitely shaped mass called pollinium. The number, shape, texture, nature of its association and associated structure vary considerably and is an important character to delimit orchid taxa at the rank of genus or even higher. This also is very presence of pollinia, the shape of the pollinia and number of pollinia also is a very important diagnostic character to come to a conclusion of this orchids. Sometimes pollinia have a stalk-like elongation known as caudicles. They will have the caudicles stalk-like elongations, which is attached to stripe or visidium. Stripe or visidium, a sticky portion will be there in the column area, which is helpful for sticking this pollinia to the body of the pollinators which are visiting the orchid flower for pollination. Visidium is the sticky pad to which the stripe or caudicle is attached by its end, carrying the whole pollinia. This helps in pollen transfer by the sticking to the back of the visiting insect. Shape of pollinia vary from globose, ovoid, and discoid to triangular and clavate. Number also varies from 2 to 8 numbers. Then pollinia is in the form of pollinia. The pollen are in the form of pollinia, which are small clumps that contain thousands of pollen grains mashed together. Orchids have 2, 4, 6, or 8 pollinia. And another important character of this orchid plants is production of these capsules. And these capsules will have millions and millions of the seeds. But here the very uniqueness is the germination rate of this orchid seeds is only 1%. The seeds are very simple. They are non-endospermous seeds. They are having the very hard testa of few cells, and in the center there will be an endosperm of 100 to 200 cells. And only 1% of the seed germination can be seen in a capsule dehisced and showing the dust seeds. They produce dust seeds. There can be up to 3 million seeds in a single orchid pod. So this is the look of a orchid capsule opened. And I showed diversity. Orchid shows uniqueness in their diversity also. I showed the very small orchids in the form of Oberonia affinis, in the form of Bulbophyllum minutissimum, to Teniophyllum alvizii, which is very difficult to locate in the forest when we are going for collection, and to very small orchid in the form of Eria excellis, Eria nana, which are very Eria nana is endemic to Western Ghats, but still very small and very much stick to the stem of the trees. And to locate it itself, you have to just open our eyes closely and just have a look. Then only we can locate this Eria excellis, Eria nana, and this Teniophyllum alvizii, Bulbophyllum minutissimum, very small, which with regard to the size is concerned. Coming to Grammatifilum speciesism, this photograph I collected from Tropical Botanical Garden, of course the center where Dr. Satish Kumar is working, where at the time of my visit to TPG area, I collected this photograph. Grammatifilum speciesism, a robust orchid in which the pseudobulb is 2 to 3 meters in its size. So then coming to conservation side, Botanical Survey of India is having 2 important orchidariums in our country. One is at Shillong, which has been established in 1959 and in conserve the orchids wealth of northeastern India. In south, we are having National Academy Excellent Garden at Yerkad, which is situated in Sanyasimale Hills, Sanyasimale forest of these Shavanan Hills, and it has been established in the year 1963 with an area of 25 to 30 acres. It is situated on the top of Shara Hills of the Salem. 35 kilometers away from Salem, we are having this Yerkard National Arcadium and Experimental Garden. At National Arcadium Yerkard, about 200 species belonging to 60 genera are well conserved. And the very important unique aspect of National Arcadium Experimental Garden Yerkard is the mass cultivation of epiphytic orchids in terrestrial beds, especially in the form of Silogen breviscapa, Cymbidium alifolium, Dendrobium aequium, Dendrobium fimbriatum, of course, North Indian orchid. Some of the North Indian elements also has been conserved in National Arcadium Experimental Garden in the form of Epidendrum radicans, then Dendrobium fimbriatum, Dendrobium cassianum, Dendrobium chrysanthemum, Dendrobium farmeri. Some North Indian species also has been conserved in National Arcadium in another section. And of course, the South Indian orchids in mass cultivation has been carried out in some of the terrestrial birds. That is one of the very important feature of this National Arcadium at Yerkard. And then we are having area pseudoclavicalis, area passiflora, area bambusifolia. All these things has been conserved. I'll be showing some of the photographs and the list of these orchid plants which has been conserved in National Arcadium and Experimental Garden Yerkard. So these are some of the collection, the collections which has been made in Munnar. I told the Bulbophyllum fuscopurpureum on the rocky surfaces, loads and loads of lorry loads of Bulbophyllum fuscopurpureum has been seen in some of the tea estates of some of this Munnar area of this Kerala. We have just seen some of the Bulbophyllums growing in so lump sum. And here is the area passiflora on some of the Western Ghat Nilgiris, especially in the Nilgiri. If you go into the deep forest, we will have area passiflora growing on the. This here is the Dendrobium herbaceum, which is a very common element. And this tree itself is having 5 to 6 species of orchids completely studied on these tree branches. Then this is in Naduvattam area of this Nilgiris. We have just made some of the. These are some of the collection sites of Idukki area I just shown. Then other collection sites, Mukriti Dam, then Mukriti area also is very, especially Bangitapal area is very good in some of the orchids. Then we have carried out work on Naduvattam area, Valley area, Nilambur, which is the border of Tamil Nadu and Kerala. Then we have also carried out work on Kautalam area, Kautalam of this Tirunelveli district, and Mukriti catchment area also we have surveyed for orchids. And some of the wild orchids collected, especially Vanilla vakari. I told another wild species of vanilla endemic to Western Ghats, endemic to South India is Vanilla vakari, which has been collected from Chitteri hills of Dharmapuri district. And then that area is also full of this Vanda tessellata species. Then in the Western Ghats, we have collected Dendrobium jordonianum. One Jayaraman, very well versed in climbing the trees, he is climbing to collect Dendrobium jordonianum from this tree. Then I am here with showing some of the lateral details which have been collected in the field, especially in the tour of Idukki area. We have surveyed Kantalur, Marayur, Namakad, Taliyar, and their lateral details also has been shown here. Just how the information, the data is collected like this. Just then further, even a particular area, their elevations, their lateral details, especially Nadugani area of the Gudulur area, we have just surveyed a lot in those Nadugani, where a gene pool of forest, Tamil Nadu Forest Department also is there. They are also having a collection of orchids in this Nadugani area. We have surveyed Naduvattam, Anumapuram, Paikara Falls area also we have just surveyed. Ovali, Kullapalli Shola, Deva Shola, all these things we have just surveyed. And further continuation of this lateral details, especially for some of the rare species what we have collected, for especially we have Vanilla vakari, which has been collected from Valiampalli of at Chitteri hills at the elevation of 3,662 feet. Then Hebinera ovalifolia we have collected at Karkampatti of Chitteri. Then Vanda tessellata, Tol Tuki of Chitteri. Then Dendrobium ovatum from Nadugani of Nilgiris, Bulbophyllum nilgirians. Then Vanda Twaitsi, area albiflora, and their areas and their lateral details has been presented. Then how these plants which are brought to the National Academy Experimental Garden, how they have been conserved, and here are some of the ways where we have just conserved some of the rare this lady slipper orchids has been conserved in mist chamber as the glass house which has been there, and you can see some of the flowers of this Papapillum spicerianum and Papapillum insigne has nicely every year they are flowering, and they have been kept in mist chamber under lock and key situation. Then we are just different methodologies have been followed to conserve these orchids in the form of hanging in the pots, And moreover, to National Archidium, many of the visitors also throughout the year, many of the visitors also coming to National Archidium just to beautify the area. Some of them has been displayed in some of the parts, as well as different methods has been tried to tie it in on the bamboo plants, and even orchid growth on the fern stem, especially this tree fern. The tree fern of this Western Ghats is the dried stem of tree fern is a very good substratum for the growth of these orchids. Then orchids in beds also, it told the beds has been also been formed. Some of the photographs I'll be showing in future slides also. Beds also been formed for this conservation of these orchids. Apart from that, we are having tissue culture facility also in National Academic Experimental Garden, where some of the tissue culture propagation, micro propagation also has been attempted. About that, at the end, I'll be at the end of my talk, I'll be just sharing some of my experiences with regard to tissue culture of this orchid plants. And these are some of the different houses which has been made in the National Archidium and Experimental Garden for conserving cymbidiums, especially for cymbidium. Cymbidium house is there, Dendrobium house is there, where some 10 to 12 species of Dendrobium has been conserved. Then coelogynes, 10 species of coelogynes are also has been conserved. Areas has been conserved separately in different sections. We are having whatnot, and then other important epiphytic orchids has been conserved in different houses. And these are the further methods of this different methods of cultivation. Here, bamboo plants, especially this method of 1 feet of bamboo plants for bulbophyllum species is very nicely. Bulbophyllum species, they just survived so nicely. Here is the Coelogyne brevis kappa growing on the one tree stem, which is a dried and dead tree stem. Very nicely, the mixture which was used here, and the plant of this Coelogyne brevis kappa so happily growing. Here is the stem of this tree fern on which this coelogyne cristata is very happily growing this here in this National Archidarium. And especially the methodology followed here is we use 3 types of mixtures for this for the growth of these orchids. And one very important aspect here is if the orchids are a little bit sturdy, the composition of this brick and charcoal will be increased. If they are smooth and little delicate orchids, there will be the leaf mulch and the loamy soil will be mixed, and this is the methodology which is followed. Even for raising some of the mass cultivation, also for raising some of the beds, also this has been followed. I will be showing in next slide. So these are the beds where some of the Dendrobium species has been cultivated in this terrestrial beds. Here is the Coelogyne vivalis. It is growing in the beds of this season, and of course this is in mid timber. The Papapedalum spicerianum is happily growing. Apart from beds, even in the different parts, also in different accessions, also in National Archidium, the plants are conserved. In the orchid plants has been conserved. And I told mass cultivation is of the epiphytic orchid in terrestrial beds is for the methodology followed is stipulated areas dig to about 50 centimeter, and the soil is scooped out. Broken pieces of earthen parts and bricks are filled to about 5 centimeter. Sometimes stone pebbles are also used. Over these, leaf mulch and pieces of tree fern, bark pieces and charcoal in 1:2:1 proportion are evenly spread over the area in a way to provide sufficient aeration space to the roots. For this, on this, some of these epiphytic orchids, the roots of these epiphytic orchids are fixed and little bit covered in the base, and they will made to grow. And in alternate days, the watering is being done, and they survive and they multiply. Especially manure is not applied, even if in dire cases, diluted cow dung slurry can be very useful for these epiphytic orchids for mass cultivation in terrestrial beds. Insecticides are also very rarely used. However, neem leaf extract or powder is very much beneficial. If any disease we found, we can very well use the mild extract of this neem leaf care powder can be very well applied for this orchids for the survivability. And now coming to orchids which has been collected and conserved in National Archidium. Here is the list of terrestrial orchids which has been conserved. And this is Acanthopium bicolor, and a very rare Enhenria rotundifolia also collected from this Kodaikanal area and Ooty area. Anectochus celatia, the jewel orchid. The leaves of the Anectochus celatia are so attractive. They will have these golden striations on the leaf, and the flower is known as white ant orchid. And then we are having Calanthe masuca, a very common orchid in Western Ghats, Cranthe triplicata, Callistus flabellata, very small ground orchid. Then Eulophaea epidendria, in which the inflorescence is 3 to 4 feet. And then Eulophaea, this we are having Epipogonium roseum, a saprophytic orchid, Geodrym densiflorum, Hebineria longicorniculata, Hebineria longicorna, Hebineria multicordata, Hebineria roxburghii, Liparis artopurpurea, and then Melaxis reedii, Nervili aragona, Thynia bicornis, the duck orchid which is called, that is also is conserved. Then we are having Papillum drury, and then we are having Thunia alba, Vanilla walkeri, Zixen gracilis, and Zixen languidatus. How these plants they look? Some of the photographs of these terrestrial orchids for better understanding I have just shown. Here is the Acanthopippium bicolor, which produces jug-like flowers and very attractive, colorful jug-like flowers. Acanthopippium bicolor, which is collected in Sheraton Hills as well as in some of the areas of Kodaikanal. This Acanthopippium bicolor has been collected. This is the Anhenria rotundifolia, which has been collected. A rare rarity species of this Anhenria orchid, ground orchid. Then we are having Anectochilus elatior, where the golden illustrations on the leaves are, that's why it is the name jewel orchid has been given. And this is the inflorescence of this produces 15 to 20 centimeter elongated inflorescence from the center of the leaf and produces this 3 to 4 flowers. Here is this Calanthe masuca having a divergent lobe in the lip, and the white flowers with violet coloration, very common orchid in Western Ghats, Calanthe masuca. Then this is the Eulaphia epidendria having 1 to 2 meters, this one elongated inflorescence, and this is the flower of this Eulaphia epidendria. So Eulaphia epidendria. Here is saprophytic orchid Epipogium roseum, which is having the transparent stem portion, and this one the underground bulb is there, and this is the Epipogium roseum. Geodorum densiflorum, which appears as soon as the rains stop in the Western Ghats. Full of Geodorum densiflorum can be seen in Western Ghats areas. Then we are having Hebineria, of course, a very big genus. We are having Hebineria longicorniculata, in which the spur is more than having a length of more than 10 to 15 centimeters. Again, similar one of the orchid of Hebineria species of Hebineria is Hebineria longicorno, in which only 10 to 15 centimeter, only 4 to 5 centimeter spur is seen. And here is the Hebineria multicordata, in which the flower looks like green orchid. And here is the Hebineria roxburghii with rosette leaves and which are attached to the soil. Then here is again this Hebineria oelifolia with green spider-like appearance. The flowers has been just picked, put on the cloth, and they have photographed in the form of this green spider-like structure. Then we have Liparis whitii and Liparis whitiana again, which is a endemic to Western Ghats, endemic to Western Ghats. Liparis whitiana, a rare entity in Liparis reticularia, Liparis elliptica, they are all common in Western Ghats, but Liparis whitiana collection is a little difficult. We just located Liparis whitiana in Western Ghats. Then we are having Liparis artopurpurea, in which the flowers are fan-like structure, and they will have this violet coloration, and this is also, even it is present in Sheraton Hills, Liparis artopurpurea. Here is the Thynia bicornis, a ground orchid in which only one leaf is present, and the flowers will look like duck orchid. The presence of these two purple structures in the anther area is the very diagnostic feature of this Thynia bicornis. Then we are having, of course, a very rare possession of this South India Papillum drury collected from Augusta Mary Hills, in which the petals and sepals will have black median band on the petals and sepals, a very rare entity of this South India Papillum drury. Apart from drury, the remaining 5 species, of this lady sleeper orchid in North India, Papillum insigne, Papillum villosum, Papillum hirsutissimum, and Papillum faireanum, they are all present in North India, whereas in South India only one species, that is Papillum drury. Then here is another Vanilla walkeri, in which we can see some of the capsules. Some of the parts of this Vanilla walkeri also has been photographed. In the Chikkari Hills, we are having this Vanilla walkeri, very of this Dharmapur district, very commonly in the deep forest. It is completely growing. Of course, Vanilla planifolia, we know it is referred to as green gold, and it is the source of this vanilla, the ingredient which is used in the ice cream industry as in the food industry. And here is the artists give us the vanilla, one of the world's most popular flavoring agent obtained from this Vanilla walkeri, in which artificial pollination is carried out, and it is referred to as green gold in horticulture as well as the agriculture field. So this is the photograph of this. Then we are having terrestrial orchid continuation, Zixine longilabris and Zixine gracilis, rare entity, especially the Zixine longilabris is commonly found in Western Ghats, whereas Zixine gracilis is very, very rare. We have located this in Western Ghat area, especially in the Y3 forest of this Kerala. We have located Zixine gracilis. Then Gooderia procera, it is a close-up is Gooderia Chirostilis flabellata also very commonly found in some of the deep forests of the Western Ghats with green tip in a green spot in the lip. We can really have, there is a diagnostic character for this Chirostilis flabellata. Then coming to epiphytic orchids, we have collected many of the epiphytic orchids from different parts. Some of them, maximum of them are endemic to South India, especially Bulbophyllum aureum, endemic to South India. Then Bulbophyllum fuscopurpureum, Bulbophyllum fimbriatum, Bulbophyllum fischeri, and then we are having Bulbophyllum tremulum. Maximum species of Bulbophyllum and Coelobane has been collected for conservation in this Western, in this National Aquarium and Experimental Garden. Even Eridis species, Acampia primorsa, which is commonly called as tiger orchid, also has been collected because of the blood red striations on the petals and sepals. I'll be showing some of the photographs of them. Of course, then next coming continuation, we have collected Catonia peduncularis, and then we have collected Dendrobium aqueum, quite bigger flowers. Dendrobium aqueum, which is endemic to South India, has been collected. Dendrobium crepidatum, Dendrobium fimbriatum, then Dendrobium herbaceum, Dendrobium microbulbon, and Dendrobium nonum, Dendrobium ovatum, and Diplocentum ricorum. Of course, it is a very common orchid in complete Nilgiris with semi-terrate leaves and pinkish colored, light pinkish colored flowers, Diplocentum ricorum. And then we are having area bambucevalia, area nana, passiflora, area whatnot, area polystachya has also been collected, and then area pseudoclavicalis. Then Flickingia nodosa, which is again a very common orchid in Western Ghats, also has been collected. Then Gastrocilus aqualis, Piparis elliptica, Lucia zelanica, especially Lucia evangelini. I will be showing the photograph of evangelini, which is a very entity that also we have just collected. Lucia zelanica is very, very common. Then Lucia macrantha is also very common, but Lucia evangelini is very, very rare. That also we have just collected. Then Oberona brunina, again endemic to South Indian, very common in Western Ghats, Brunoniana. Then we are having Oberona verticella, has also been collected. Then we have collected Papilanthes subulata. A very, very aromatic smell comes from the flower of Papilanthes subulata, and Papilanthes terris. Papilanthes terris, which is called butterfly, which resembles the flower resembles the butterfly, and Papilanthes terris also has been collected. It is again very common in all parts of Western Ghats. Polydendra imbricata, which is referred to as rattlesnake orchid, because the end tip of the inflorescence of this Polydendra imbricata resembles the tail of the rattlesnake. And then we are having very common Polystia concreta also. And then the Porpax jardoniana, which is commonly called button orchid, also has been collected. 2 species has been reported, Porpax jardoniana and Porpax reticulata. We happen to collect Porpax reticulata. And then we are having the Rhynchocelis retusa, of course, the foxtail orchid. Rabecacia gracilis, again a rare orchid, has been collected. Rabecacia josephiana also has been reported. Sedum fenella chrysantha also collected. Sarhocaria lanceolata, a very small orchid growing on very, very big trees. Sarhocaria lanceolata also has been, lanceolata and latifolia 2 are there in Western Ghats. Then Tinephyllum alvizia, I told, leafless with assimilated roots, very small orchid, Tinephyllum alvizia. Trias toxia, again endemic to South India, has been collected. Then we are having Vanda testacea, which is very common in Western Ghats. Then we are having Vanda tessellata. Vanda spatumata, also little rare in Western Ghats, and that also has been collected. And Vanda thoitsi, the first distribution record to Tamil Nadu, we have reported, myself and Dr. Murthy Saab has reported Vanda thoitsi, the first Tamil Nadu record. Of course, Vanda thoitsi has been reported from South India by Dr. Satish Kumar. And Genicophyton smianum, a rare species and endemic to South India, also has been collected and conserved in South India. How they look like, some of the photographs. And this is Eridis odorata, and a very common character is the peacock character in the lip. Lip is the very important character of this Eridis maculosa. Then we are having Eridis ringels. 2 varieties has been found. One is white variety and pink variety. Both of them has been collected. Acampia primorsa, the tiger-like striations will be there. So Acampia primorsa and Acampia acrosia also is present in this Western Ghats. Then coming to Bulbophyllum, I told again a very big genus Bulbophyllum, a very, very big flower of Bulbophyllum, especially this is Bulbophyllum fuscopurpureum, with having very good modification of hair-like structure in the flower, that has also been collected. And this is Bulbophyllum aureum, the single flower with white or yellowish white color. This is bulbophyllum mysurans, and then here is bulbophyllum fishery, which is having umbellate flowers of umbel of the flowers of 5 to 6 this one flowers. And here is bulbophyllum fimbriatum. Here is bulbophyllum nilgirias and bulbophyllum tremulum. Here there will be appendages in the lip area and which are mobile. If you just see it from the close angle, 90-degree movement with small breeze, 90-degree movement will be very attractive in this bulbophyllum tremulum, which is again endemic to South India. And then continuation, we are having this Coelogen breviscapa, again endemic to South India. Coelogen odoratissima, very excellent and odorous. We collected this and we are staying in the guest house of the tea board completely for 8 days. Complete room was filled with full fragrance, especially this Coelogen odoratissima, again endemic to South India. Of course, in Mukriti National Park, also in the Bangitapal area, also this is fully commonly present. We collected this for the first time in Nadotam area of this Nilgiri Hills. And here is the Coelogen nitida, and here is the Coelogen nervosa. Especially the nerve-like striations on the pseudobulb is the very important digestion character of this Coelogen nervosa, again endemic to Southeast Asia, so endemic to Southwestern Ghats, and very common in Devikulam area, Devikulam and Marur area of this Kerala or the Idukki district. This is very common, and this is Coelogen nervosa. And here is bull's head orchid, Coelogen ovalis. Coelogen ovalis again in Western Ghats, very common and very rare. Very common and very important and very attractive orchid called bull's orchid, Coelogen ovalis. And here is Cymbidium alifolium or Cymbidium bicolor, and of course this is North Indian orchid that also you have considered, Cymbidium giganteum. Cymbidium giganteum. Here is Cymbidium bicolor, and one more very common little white is Cymbidium aloifolium. And here is some of the Dendrobium aequium, very important, attractive white flowers, greenish white flowers coming from the axils of the leaves, and all over the surface of the pseudobulb we get this Dendrobium aequium. Here is Dendrobium macrostachyium and Dendrobium crepidatum. From the axil of the pseudobulb, some of the flowers, pinkish color, whitish pink color attractive flowers comes up. Dendrobium crepidatum. Here is Dendrobium fimbriatum, of course, a North Indian orchid fully established in NYG ERCOT, and one hill hillock itself is completely full of Dendrobium fimbriatum, and some of the visitors, this plant has been sold for this visitor Dendrobium fimbriatum. In the month of March and April, if you visit National Arcadium, it will be fully golden yellowish color, and it will be fully conserved, fully developed, multiplied in the National Arcadium. Here is Dendrobium whitei, again RET, a very rare and endangered species of this Dendrobium present in Western Nilgiri area, Dendrobium whitei. Of course, very small white, greenish white flowers, Dendrobium herbaceum is fully commonly found in all parts of the Western Ghats. And then here is Dendrobium heterocarpum, golden yellowish colored Dendrobium heterocarpum, endemic to Western Ghats. Here is Dendrobium anili. The greenish portion in the lip of the plant is very distinguishing character. Here is Dendrobium ovatum, creamish yellow flowers, and again common in many of the Nilgiri areas. Here is Dendrobium nanum, very stunted growth of the plant is seen, and small pseudobulb will be present. This is Dendrobium nanum. And then this is the Diplocentrum ericarum, I told, very commonly found in all Western Ghats as well as Nilgiri area. We have got semi-terete leaves of this Diplocentrum ericarum, and the flowers are pinkish white, attractive flowers. And coming to the area species, again, area is also very good. Population of area has been seen in Western Ghats. One is area bambusifolia. The pseudobulb is little elongate, 1 to 2 feet growth is there, and it is having constrictions as that of bamboo, hence the name area bambusifolia. Red striations on the complete flower can be seen, area bambusifolia. Area pseudoclavicalis, very again endemic to South India, this Western Ghats, and very attractive flower with violet striations and violet patches, and very attractive. Even the single flower, 1 to 2 flowers come in the axil of the leaf, 2 leaves, so very attractive flower of this area pseudoclavicalis. Area polysteca, again endemic to South India. And then area nana, very small orchid and having star-like flowers, 3 to 4 flowers they come up, and very small flowers, again endemic to South India. Area nana. And then area albiflora, again very small orchid. It will have a exact very important constriction in the in the pseudobulb is in the exactly middle of the pseudobulb. There is a constriction, that is a diagnostic character of this area albiflora. And then coming to other epiphytic orchids, this is Polystachya concreta, again common orchid in Western Ghats. Flickingeria nodosa, the close up of the flower of this Flickingeria nodosa, very common, and on the stem of the big trees, this Flickingeria nodosa will be completely growing in this Western Ghats. And this is Papilantha terris, I told, butterfly-like orchid, and this is the Cleistostoma tenuifolium. Here is the diagnostic character is the presence of human tongue-like structure appendage in the flower, and that is the diagnostic character of the Cleistostoma tenuifolium, a rare orchid encountered in Western Ghat area. Then here is Liparis elliptica, and this is Liparis bridiflora, like ants in line. If you see the inflorescence, we just see as though the ants are just moving in the line. So Liparis bridiflora. This is this rattlesnake orchid, Polydota imbricata. The tip portion of the inflorescence looks like the tail of the rattlesnake. This is the mouse tail orchid, Oberonia brunoniana. This is in bud condition, and this is in bloom condition. Again, endemic to South India. And here is Lucia evangelini, Lucia zelanica, very common, and with terete leaves. And then here is the Lucia evangelini. I told this is rare and endemic to Western Ghats. That also we have just collected in our collections. And then we are having this Sarhucaria lanceolata, very small orchid in very big trees of this Western Ghats. Of course, no pseudobulb, and with the roots it attaches to the stem of the trees and there's grows. And here is one more exotic orchid which has become common and which has become wild, that is Epidentum radicans, commonly called as Holy Cross orchid. In the flower itself, there will be Holy Cross. Of course, when I was there in Yercaud, one person from Germany, every year he was coming and collecting the flowers of this Epidentum radicans. I don't know what sentiment had been attached to the Epidentum radicans called Holy Cross orchid. Then here is this Papilantha subulata. It told aromatic flower is only 1 or 2 flowers will be there, and completely long cylindrical leaves. It sometimes we get confused with that of Lucia zelanica, but little elongated and cylindrical leaves are pseudobulbs are found in this area. In this Papilantha subulata. This is Tinophyllum alvizei, the symmetry roots, and some people call it as snake orchid. This is Trias stocksii, again endemic to southwestern, endemic to South India. And this is Vanda thwaitzii, we have collected from Tamil Nadu area, and especially from Kodaikanal area, Vanda thwaitzii was reported. First, Satish Kumar has reported this from Kerala. Of course, there was no distribution record for Tamil Nadu. We just reported it as first distribution record to Tamil Nadu, Vanda thwaitzii, with creamish yellow flowers with purple striations. And then Vanda testacea, of course, very common orchid in Western Ghats. And then Trichoglottis tenera, again very rare and endemic to South India. The book written by J. Joseph, The Orchids of Nilgiris, the covering pages of this Trichoglottis tenera, the diagnostic character is all throughout the stem of this plant. It produces wiry roots. Wiry roots, the presence of wiry roots on throughout the stem is the diagnostic character of this Trichoglottis tenera. This I will be showing the figure of the orchids of Nilgiris. And here is the Porpax reticulata and Rabucacia gracilis. He is the Rabucacia gracilis, again rare entity, and the only monogenic species of South India, Zenicophyton smianum. Coriaceous leaves are present, and from the axil of the leaves, the inflorescence grows. And this is the close-up of the Zenicophyton smianum, again endemic to South India. This is Porpax reticulata, button orchid, what we call, and here is the flowering. Trumpet-shaped flowers are present, produced by this Porpax reticulata. So this is the Rincochalis retusa, the foxtail orchid, luxuriously growing in some of the trees, especially in this Nilgiri areas. And this is used for ornamentation of the hairs by ladies in North India. North India also this distribution is there, this Rincochalis retusa. And apart from that, apart from carrying out work on orchids of South India, when I was holding the charge of National Orchidarium, after in 2013, in the midst of 13, when I got transferred to AJCB Indian Botanic Garden, I took a project on collection and introduction of ex situ conservation of rare and endemic orchids in northeastern India. for at least for 3.5 years, I worked on these orchids of here. Some of the, just to know the glimpse of this, the difference between the elements of South India to North India. Here is Epigenium amplum. Some people call it as moth orchid. Tiger orchid, Esmeralda cathcarti, very beautiful Esmeralda cathcarti. This is Pleon perax, very beautiful and attractive orchid, Pleon perax. Of course, blue Vanda cerulea is also very common in northeastern India. Some of the species we have collected here. I just this one Iridis odorata, then Agrostophyllum callosum, Bulbophyllum carianum, Phyllocista parishi, Silogin cristata, Silogin fuscescens, Dendrobium nobile. Of course, this is the state flower of Gangtok, Dendrobium nobile, very beautiful and very attractive flower. The photograph I'll be showing in next slide. Dendrobium devonianum, Epigenium amplum, Esmeralda cathcarti. This is Esmeralda cathcarti. Nervilia macroglaza, a rare terrestrial orchid, also has been collected in this northeastern India. And these they were somewhere acclimatized in this National Aquarium at AJCV IBG. Here is Dendrobium longicorno, which has been just flowered in this National Aquarium. First acclimatization, later shifting to orchidarium. These are some of the photographs of this rare orchids collected in Northeast. Dendrobium nobile, a very beautiful flower of this state flower of Gangtok, Esmeralda cathcarti close up. And then Vanda stangini also has been collected, and Nervilia macroglaza, a rare which resembles that of Epipogum roseum, and this also is collected in northeastern area. And some of the publication in this orchids line, Survey and Conservation of Rare and Endemic Orchids of Western Ghats, published in Journal of Orchid Society of India, which was at that time the president was Professor S.P. Vij, and then Vanda Throichi, the first record for Tamil Nadu, has been published in that one. And then a novelty has been reported as Bulbophyllum paramjiti from North Sikkim area. We have just collected this one, and that has been reported in Phytotaxa. Then I presented this work in second Indian Biodiversity Congress held in Indian Institute of Science, Bangalore in 2013. And this is the preceding volume in which Orchid Diversity of Western Ghats has been published. So these are some of the publications which has been brought out in these lines of this orchid conservation. And these are some of the books which are very much useful in the lines of the orchid culture, orchid study. This is the Orchids of Nilgiris by Joseph, and this is the Trichoglaptis tenera. The photograph of this Trichoglaptis tenera has been shown as the covering page for Orchids of Nilgiris. Even though the book is small, but very useful, especially for identification of South Indian orchids. And here some of the other orchid related books. Of course, here one recently brought out by BSI, Orchids of India by S.K. Singh. I have just not put some 1 year back. Orchids of India by S.K. Singh also has been published by BSI. So these are some of the books related to orchids. And then come to the last aspect. Now time is also running. Coming to last aspect, tissue culture of orchids. There is a facility for tissue culture in National Academic Experimental Garden. We are having a good lab of this tissue culture in National Academic. When I was there, some tissue culture also we attempted. Here, with regard to tissue culture of these orchids, tissue culture of orchids was first demonstrated by Neil Bernard, a French botanist who just demonstrated this tissue culture. Especially this tissue culture of orchid is carried out in two ways. One is seed culture is carried out, and second is mariculture or meristem culture is carried out. Two aspects are there. And since mariculture we have not attempted, seed culture we attempted, I'll just stick on to the seed culture. Again, in his seed culture, the very important aspect of what we have to know is, once the seed develops, we know orchids produce seed dust. Millions of seeds are present in the capsule. Of course, the seed germination is only 1%. Nature has not gifted these orchids with the high germination rate of this orchid seeds, because seeds are very, very minute and they are not at all having endosperm. Only seed testa is there, and then in the center a small embryo, embryo exists of 100 to 200 cells. So this seed, as soon as it germinates, for further growth of the seedling, it requires the association of mycorrhizal fungus. There only the problem comes with regard to tissue culture of the seeds, because tissue culture and fungus are completely two lines of the rail of the this one train, I can well say. If fungus takes entry into the media of this tissue culture, completely it gets dominated. So that's why here two aspects are there in this seed culture. One is asymbiotic seed germination, and the second one is symbiotic seed germination. Especially symbiotic seed germination is attempted in temperate orchids, whereas asymbiotic germination is attempted in tropical orchids. Here for this symbiotic seed germination, it doesn't require specialized media. A simple media of this oat extract is very much enough for this temperate temperate orchids for this attempting this tissue culture. Whereas for asymbiotic seed generation, a well developed media in the form of Knudsen media, Murashige Fuchs media, then we are having niche media, high media, and then Mitra media can be tried for this asymbiotic seed germination. We tried two types of media. One is the Murashik and Scoop media, and the second one we attempted is Mitra media. And especially orchids doesn't require complex inclusions in the media. Very simple compositions, half strength MS media and Mitra media, which is having lower concentration of this organis, inorganics, and this iron source and growth regulators are found to be very beneficial for propagation of these orchids. And yeah, this especially this Mitra media was very helpful, and we saw the use of 15% of sucrose for the inclusion of this media is enough.
Dr. Sharif.
Sir.
Now you wind up.
Just 10 minutes. I'm just at the fag end, sir. Just in 5 minutes I'll complete. So this is the method of this seed culture we have just carried out. And here the capsules were taken, they were rinsed, and then they were flamed in the ethanol, and then they are completely cut, and seed sowing is done. And how the response is seen, I will be showing in some of the slides. Just 5 minutes I'll be just winding up, sir. And these are some of the tissue culture works we have carried out. Here the seed culture that has been carried out, and seed germination takes place. And after germination, immediately they form PLBs, protocorm-like bodies are formed, and these protocorm bodies can be separated, and they can be tried on different shoot maturation media consisting of the half MS or Mitra media with lower concentration of growth regulators in the form of Na and BAP can be used for maturation, and mature shoots can be very well completely just obtained, and they can be subcultured on some of the filter paper bridge with liquid media, and they can be allowed for maturation. After maturation, even some of the shoots can be made to root also by the using of this some of the oxygen concentration, more oxygen concentration, and they can be very well transplanted to greenhouse in the and with the help of this vermiculite and perlite, these plants were made to establish, and they are we have just got very good success in this tissue culture, especially in the plants like Silogin nitida, Cymbidium alvifolium, and Acampia primorsa. Some of the plants has been completely propagated, and micropropagation protocol has been standardized. So with this, I think I come to conclude my topic. Indian orchids are paradoxically victims of their own beauty and popularity. It is obvious that as large orchids enjoy an economic status and demand, their exploitation from nature will continue. They are, of course, their beauty and their attractive flowers are they are the victims of their own nature. To reduce this pressure, mass propagation by using conventional as well as tissue culture techniques would be an important strategy. From last decade, Western Ghats have witnessed large-scale destruction resulting in decline of orchid diversity. Hence, conservation strategies such as in situ, ex situ, preservation, orchidarium maintenance. We have to raise many of the orchidarium for conserving some of these orchids of those in the native areas. And community efforts are also very important, because people should know what are the detrimental effects, because orchids are very good environmental indicators. So community efforts are very much needed. Without the help of the local people, conserving the orchids is very, very difficult. So community efforts assume great importance. More efforts are required to be taken for preparation of national inventory orchids, identification of rare and threatened orchid taxa to formulate effective conservation programs. In addition to this, local communities and general public should be educated about the need of preservation and conservation of orchids. Otherwise, most of our wonderful species and botanical jewels, these orchids, may get vanished forever. That's why conservation of these orchids is very, very important. And of course, the conservation of biodiversity has become a necessity rather than a fashion today. And we have to conserve these very important entities of this Western Ghats. And thank you very much. Long live Indian orchidology. Thank you. I once again thank the authorities of Krishnamal College for giving me an opportunity to share my experiences and whatever the information which was there with regard to this orchidology. Thank you very much. Thank you.
Thank you, sir. Now the participants can clarify their doubts.
Okay.
Good evening, sir.
Good evening. Good evening.
Very nice and informative presentation, sir.
Okay.
My question is, what about the roots of orchids? Because the roots having some particular character now compared with other plants.
Yes, I told they produce specialized roots called aerial roots. That is a very important adaptive character of this orchids to production of aerial roots or velamen roots. It is called specialized tissue called velamen, and that will be helpful for absorbing the moisture content from the atmosphere and help for the growth of these orchids. This is a very important. Apart from that, aerial roots, some of the orchids, these terrestrial orchids, they will have the general roots in the down, as well as some epiphytic orchids also have some of the roots in the pseudobulbs. Thank you very much. This is the information.
Thank you, sir. Then another one question. Did you any seed germination and viability studies, sir? Whether you can explain which method is best for orchid? Seed germination and viability test.
I told the orchid is the field where especially the phenological studies has not been much done, and my morphometric studies have not been done, and especially the florobiology of this orchid has not been worked out, because interior forest one has to move. There are so many restrictions. Many of the orchids are under appendix 1 of this RET plants. So much of the work has been in orchids has been it is infancy, and especially the seed germination. I told nature has not gifted orchid with high germination rate. Otherwise, many of the orchids would have been survived in the nature. Only 1% is the seed viability, and of course, tissue culture is the best one we can obtain by especially giving the concentration of this kinetin. Lower concentration of kinetin is very, very helpful for enhanced germination of the seeds can be attained. This is the one I can very well share with you.
Thank you, sir.
Thank you.
Sir, good evening, sir.
Good evening.
Thank you for your wonderful information, sir. I have doubt regarding the that is in vitro seed germination of this orchid, sir. But in vitro seed germination is very successful. But when we are go for organogenesis, so the leaves and then the stem parts like that, they are in connection with it of the endophytic organisms.
You are right. You are right. Of course, many of the monocotyledons are not good components for tissue culture. Of course, orchids also the presence of this fibrous tissue and these epiphytic orchids are very sturdy, and that's why they are not the good entities for tissue culture. We can very well select many of the species are there. Of course, 250 species are there in Western Ghats. Some of the very rare, which are very important economically, they can be selected and they can be put for tissue culture. That can be very well attempted for. Okay.
Sir, there are two questions in the chat box of YouTube Live, sir. Shall I read it, sir?
Yes, please read.
So the first question is, how taxonomical studies will help budding researchers for funding sides?
For this, the right person is Sanjay sir only can give, because he is only can be the right person for funding area. We cannot give much information, especially INSA, and even, of course, I can well say BSI also is having assistance to botanical gardens, especially if you have got basic infrastructure facility. Assistance to botanical gardens can be attempted, and you will get grant for conserving some of the rare and endangered plants in some of your botanical gardens. So otherwise, other projects are to be, other fellowships are to be attempted. These are the ways.
No, what funding you wanted actually?
Yes, sir. It depends on that.
Student, whoever is the participant is asking for funding. No, what kind of funding they want? They want for project funding.
Hello.
Sir, the question was asked in the chat box, sir. So we will let you know, sir, who is the person who asked the question.
No, we want to clarify her first. So
yes,
what is
sir.
the question exactly? She wants funding for research purpose or some other purpose?
I think it's for research purpose, sir.
Oh, there are good opportunities, but she has to compete with all other science subjects. In DSP, DBT, Minister of Environment and Forests, there are opportunities to get funding for any taxonomy projects. Only thing, they have to be competitive.
Yes, sure.
And they should have relevance, and there should be some complexes that are to be solved. Not for simply writing flora of the area, they will not give funding. Say if you take up say Agasthamala flora, maybe they will give you, and you will have to see what we are going to, in return, what you are going to give.
Okay. The next question.
Am I clear?
Yes, sir. I think that's clear, sir. Thank you, sir.
Okay.
Next, what is the next question?
What are the funding agencies for taxonomical works?
Just now, sir told MOEF is there under assistance to medical. This one, INSA is there, UGC is there, DST is there, DBT is there. They are all the funding agencies we have which are funding this project. But effective results should be given. Then only funding will be effective. Okay.
Okay, sir. Thank you, sir.
Thank you. Any other?
Akshay, I think you can proceed.
Thank you, sir. Now I request Dr. M. Kanchana, Assistant Professor, Department of Botany, PSCR Krishnamal College for Women, to welcome the speaker of the technical session 4, Dr. V. Sampath Kumar.
Good evening all. It's my immense pleasure to welcome the speaker, Dr. V. Sampath Kumar. Dr. V. Sampath Kumar, after completion of MSc degree, joined as a research scholar under Flora of India project at Central National Herbarium, Botanical Survey of India, Kolkata. He received his PhD from Calcutta University in 1997 and joined as botanist in Andaman and Nicobar Circle of BSI, Port Blair, in the same year. During this period, Dr. Kumar visited various islands and contributed for flora of the Andaman Islands. He has documented mangroves of Andaman and Nicobar Islands. After serving 4 years in the islands, he joined as scientist in Central Drug Research Institute, Lucknow. In 2002, he has joined again in Botanical Survey of India, Kolkata, as scientist. During this tenure, he continued to contribute for the flora of India for the family Lamiaceae as well as for flora of West Bengal. In 2006, he was posted in Deccan Circle, Hyderabad, and after a couple of years, he was posted in the Southern Region Center, BSI, Coimbatore, where he worked on flora of Kerala and documentation of Vellingiri and adjoining hills. In 2011, he was deputed as Indian Botanical Liaison Officer in Royal Botanic Gardens, Kew Garden, London for 2 years. He has visited several European herbaria and botanical gardens in connection with the Flora of India project. After completion of the tenure, he joined in Central National Herbarium, Kolkata. He was in charge of ENVIS Center, Environment Information System, sponsored by Ministry of Environment, Forest and Climate Change, Government of India, New Delhi. During this period, he has conducted several workshops, especially on herbarium techniques, plant identification and plant nomenclature courses to the PG students, the research scholars and college teachers. He was also a national coordinator for the Green Skill Development Program, in which the training were imparted to the students on biodiversity and conservation aspect. In the financial year 2017 to 18 and 2018 to 19, he has conducted the Green Skill Development Program by obtaining about 2.5 crore from the Ministry of Environment, Forest and Climate Change, Government of India. During his more than 30 years research career, Dr. Kumar has visited various phytogeographical regions of India for survey and documentation of plant wealth in India. He has published more than 60 papers in various Indian and foreign journals. He has described 5 new taxa and made more than 60 new combinations and new names. He has also co-authored and edited half a dozen books. He is the member of various botanical associations and societies and fellow of Indian Association of Angiosperm Taxonomy and Indian Ethnobotanical Society. Dr. Sampath Kumar is the recipient of Professor B.A. Razi Medal in 2016 for his significant contribution to the taxonomy of flowering plants of India. Presently, Dr. Kumar is working as Scientist E at Southern Regional Center, Botanical Survey of India, Coimbatore. Once again, I welcome you, sir. Thank you.
Thank you, ma'am. Now I call upon Dr. V. Sampath Kumar, Scientist E, Head, Southern Regional Center, Botanical Survey of India, to deliver his lecture on the topic Botanical Nomenclature.
Sir, please unmute yourself, sir.
Good evening to all. Am I audible?
Yes,
Yeah,
sir,
yeah.
you're audible.
1 minute, please. I think slides is visible.
Yes, sir, it's visible, sir.
Yes. Okay. First of all, I thank to convener. Actually, Sanjapa sir is my mentor. When I joined as a research scholar, I learned whatever taxonomy I know from Sanjapa sir only. I have learned. That is, I am thankful to him. I am also thankful to Principal PHR Krishnamal College, Head of the Department Krishnavani Madam, and all other organizing members, including Dr. Tamil Chelvi. I am thankful to them for giving this opportunity. Actually, I have prepared this slide for basics for the students only, with a brief history and small basic only. I am going to, it is mainly prepared by keeping in motto of the research students only. Is it slides moving?
Yes, sir. Yes, sir. They are moving.
Yeah. What is taxonomy actually? Taxonomy is nothing but identification, nomenclature, naming of the plant and classification. If it is only to plants, we refer as systematic botany. Once the plant is identified, it is necessary to give the name, scientific name. Botanical nomenclature is nothing but scientific naming of plants. It is related to but distinct from taxonomy. Please note this word. It is related to but distinct from plant taxonomy is mainly concerned with grouping and classifying them. Naming of plant is a function of taxonomy regulated by certain rules or code in botanical nomenclature. These rules are direct the procedures to be followed for the determination of the name to be applied to a particular plant or to be provided a new name for a plant. First of all, many conferences and many persons they asked this question: what is the difference between nomenclatural and nomenclatorial? When the subject is nomenclature, that means the naming of the plant, the adjective is nomenclature and the adverb is nomenclaturally. Suppose a compiled book is there, whatever the names and the synonyms are compiled, that is called nomenclatorial work. So I think everyone knows the index sequences. Everyone seen whatever the throughout the world up to I think 1980, whatever 1980s, whatever names published, that is compiled in the index sequences. It was started by Hooker. It has been started from Q. It has been published from Q, that is Royal Garden School, that is called index sequences. Why scientific and botanical names are required? For, as you know, for the different vernacular names are available in different regions or in the same language itself. For you see, in India itself, for paddy, Orissa sativa, you call it in different names. In Marathi, they call Batta, Kannada also Batta, and BM in Telugu, Nel in Tamil and Malayalam, and Dan or Dana in Hindi, Nepali, Bengali, everything. That is why different places differently they call. That is why botanical name is required. Even in the same language, same plant is known in different place. Suppose you take this Cucurbita pepoaceae, this. You can say in English itself it is called in different names. In Hindi, Kaddu, Kasifal, Kumra, Sitafal, etc. they call. On the other hand, same vernacular name is to wholly different plants. Because Sangupushpi, it is known throughout India, Sangupushpi is called for different plants. For different families it is applied. Bacopa moneri, Canscora diffusa, Clitoria ternatia, Convolvulus pluricalis, and evolved suchnoids are called as Sangupushpi. Same way, Ashoka tree. In South India, we call Polyalthia longifolia as Ashoka tree, but it is originally Charaka Ashoka is the typical Ashoka tree. Polyalthia longifolia, because the Samrat Ashoka has planted this plant, that is why we call it as Ashoka tree. See, this bird of paradise is Sterlitzia reginea. It's a monocot from the tropics, it is there. But in North America, in some desert plants, Cisalpina gillesi, Fabaceae leguminaceae plant is also called as bird of paradise. See, Indian hemp, here I'm giving some examples. Different genera it belong, but India, these hemps are called different names. You can see here. For Apocyinum, Cannabinum, and Hibiscus cannabinus, Asclepias incarnata, all are called as Indian name with a different prefix they are having. For gardener also, it is easy to use botanical name rather than a common name. It is very easy. We can say scientific names do indicate generic and usually genetic relations. Scientific names is botanical names. Hence, they are biological of significance we have. And we can say for the common plants only vernacular names are available. Uncommon plants, we don't have any vernacular names. Every plant must have only one correct botanical name should be there. The earliest, that is in accordance with the rules, rule of priority, that I will come later. For common ginger plant, for common ginger plants, Gingiber officinal is the accepted name. All these three are synonyms. Maybe more synonyms. Here I listed only 3 synonyms only. That is, I think, hope you people are knowing the synonyms, everything also. Because the botanical nomenclature aims at the provision of a stable method of naming of taxonomy groups, avoiding and reducing the use of names of may cause error or ambiguity or throw science into confusion. Because botanical names, once it is proposed, means that will solve many confusions. For that purpose, it is there. I will give brief history of taxonomy. After that, brief history of botanical nomenclature. After that, only 5 topics I will cover. Here, taxonomic brief history I will go. The first classification of plants reported in Vedas around 2000 BC itself. That means around 4000 years before itself. Here in the Vedas, classification was there. But in text you will find the early Egyptians and Greeks named many food plants and others with medicinal properties. One of the most important early works was the Theophrastus in the 3rd century BC, who recognized the difference between the different plants, what now we call monocots and dicots. This Dioscorus, Pedarius Dioscorus, a Greek physician of the 1st century AD, who wrote mainly about medicinal plants and who recognized groups, what we call them as a families. The Romans imposed their civilization on very large area of the known world of that time. Their influence of their language, Latin, became the way of laymen communicated with each other. Many present-day plant names stem from Roman times itself, including the common names also. Initially, a single name would serve the purpose. Say, rubus means the preamble, it will serve. But later, for different species, they wanted to add that became a polynomial, the preamble with the large leaves and black juicy like that. Practically saying, this botanical progress was not much until the 16th century, when a number of useful books printed mainly on herbals in the 16th century. But in the 16th century also, the importance of the flowers and fruits remained unrecognized only. Actually, everyone knows binomial system is introduced by Linnaeus only, but actually it was first used by in the 17th century, 1620, by Swiss botanist Caspar Bogin. All you know the Boginia plant you are knowing. Based on his name only it is coined. Caspar Bogin used the concepts of genus and species. But unfortunately, he has not used consistently. And this Rudolf Jack in the 17th century itself, yes, Rudolf Jacob Caverius was the first to recognize the sexual nature of the plants. That is, flowers and stamens, gynece, everything he was introduced. In 1753, Linnaeus published a book whose title is, usually we call it as Species Plantara. This written in Latin as was the custom of the time. There he used the binomial system. It is called conspectus because some brief description, it is not a nomenclature. Because what nomenclature I told, that is name simply indexing of the plan, whatever the names available that time, either accepted name or synonyms. Linnaeus, who Latinized his name as Linnae, published a large number of plant names with detailed description each. Yes, published. Actually, it is considered valid. It must first be published with a small description at least. Description in Latin they have made. That time they used to call it as described, which was accepted by botanists of any nationality that time. But in these days, this problem in these times when few botanists only know the Latin. Why botanical names are based on the Latin language? Because Latin language is specific and exact in meaning. It is very precise and conciseness. It is expressed very clearly. Suppose on name itself given means it will indicate what is the plant. This some characters we can assume that. Yes, already I have mentioned this Latin was language used for communication between the scholars in Europe that time. That is around 17th and 18th century. Even it was no longer an everyday language it is there. See, Kerala means a little crown mean, but it has been used for the chapels as you know. And similarly, before the middle of 80s, names of plants are commonly polynomials only. Until 1753, although 1620, Caspar Borgen used the binomial. See, this Ranunculus bulbosus, they used this big name, Ranunculus calycipes, retroflaxis, something, and compositus. This much. In this itself, small description is there about the plants it is described. The binomial system postulates that every species consists of only two words. The increasing number of new plants caused concern over methods of naming the new names or novelties. Whatever we are knowing, this naming of the plants, it is beginning in Linnaeus Critica Botanica in 1737 it was. It was primarily to explain the taxonomic and the nomenclature principles employed by Linnaeus in his Genera Plantarum and Artes Cliptonius. In 1751, later before publishing the Species Plantarum, Philosophiae and Botanica, Linnaeus amplified the views expressed in his aphorisms and principles and established the real meanings of the sound nomenclature of the plants. After that, in the next century, the theory elementary della botanica was published by the Augustine de Condor is the first on exploring matters of organography and nomenclature. Because in the starting of 19th century, they have made this. He gave explicit instructions on nomenclature procedures, many of them taken from Linnaeus itself. Although de Condor did not use much modern words like epithet, homonym, tautonym, nomonym, etc., but what he was meant for that. Again, after 1830, in 1821, Stuart's Nomenclator Botanicus was first important post-Linnaean index to the names were published about the flowering plants. And in the second edition in 1840, he has a detailed account, whatever the names available that time, he has published either accepted or synonyms. These are the actually forerunner of index coincidences, or this nowadays what we call in this what is this internet era, IPNI, Tropicos, the plant list and all the basis for this the students nomenclature botanicus work on. I come to brief history of International Botanical Congress. The first International Botanical Congress was held in Paris in 1867, headed by Alphonse de Condor was organized. Of course, in this very limited persons, Europeans and Americans only participated. There, the laws were recommended as the best nomenclature in the vegetable kingdom. Also known as de Condor rules or Paris Code, it was there. This is starting point for all plant nomenclature was with Linnaeus. Although it was not enforced fully, because this most of the American botanists, they have not accepted, especially they are having this Asa Gray, like that peoples, they were following their own set of rules in America. That is why it is not fully implemented. See, just I have taken from the IAPT website. These are the Botanical Congress, whatever it is held. Although I told 1867 was the first it was held, but it is not. As per IAPT site, it is 1900 once the first Botanical Congress was held. And until 1950, the Stockholm Congress, it was called as International Rules of Botanical Nomenclature. After that, in the Stockholm 1980, first International Botanical Nomenclature as ICBN 1950 was published. Again, from 2011, after 2011 as 12th and 30th international, this name again, ICBN has been changed into ICN, International Code of Nomenclature for Algae, Fungi and Plants. It is effective from 2012. Yes, I already told this in Stockholm only, International Code of Botanical Nomenclature was passed. In the same year, this International Association for Plant Taxonomy was established, and the Taxon Journal, the very important journal for taxonomies and nomenclature aspect, it has been published. It was here also that taxon name was introduced. What in 1972, this in Seattle Code, they have introduced the word taxon. Taxon, it is applicable to any rank. It is applicable. It is genus, species, or family, whatever it is. That is taxonomic group. Taxon word is coined there only. Clearing some confusion. Already I have told that 1867 code. It is not that 1900 Paris Congress only first International Botanical it is accepted. Although it was held in 1867, most of the Europeans, Americans are attended, and this has been changed from rules to code in 1950s. From the 7th International Botanical Congress, Stockholm onwards, it has been changed. This last code was the Shenzhen code, was the last one, and the next in Rio de Janeiro it will be held, but in this COVID time we cannot assure that. But this is some important decisions whatever they made, that I have made this. The concept of registration of plant names, algae, bryophytes and vascular plants is accepted. That means whatever new names it is there, it should be registered. Maybe in 2025 it may be implemented. Already for mycology it is effective from January 2013. Generic name typification is proposed before and by the followers of American scores are not accepted. In the Shenzhen code, there is a separate chapter on items pertaining to mycology alone itself. It is first time introduced. Several typical committees have been set up to examine typification, DNA sequences, list of available names, pleomorphic fungi, and virtual participation in the nomenclature section. These are introduced in the Shenzhen code. It is introduced in 2017. It is published in 2018. In the 20th IBC will be held in Rio de Janeiro, but in this COVID situation, after a couple of years, whether it will be done or virtually it will be conducted, we do not know. After the brief history, I will cover basics of nomenclature in this 5 types. Major rules, that is categories and hierarchy, I will use. Typification of plant types, I will cover. The rule of priority, 2 slides, I will. Effective and valid publication, what is that? And how to cite author citation and what is illegitimate. With this half a dozen titles that is useful to the students, I will cover these things. These categories and hierarchy. You see, from kingdom to species, it is there. But here, domain word also introduced. Kingdom to species, it is there. Domain word also it is there. Actually, taxonomic hierarchy categories are also introduced by Linnaeus only. They are also known as Linnaean hierarchy. It is defined as sequence of categories in a decreasing or increasing order. Kingdom are sorry. Kingdom is the highest rank, followed by division or phylum. Although this domain was proposed by the Carl Lewis, that is prokaryotic or eukaryotic, it is there, but in nomenclature code, this domain is not used. The hierarchy has two categories which are obligate and intermediate. Obligate is the compulsory one and it should be registered for intermediate. Thus, so obligate is it is kingdom, genus, species, family, like that. Intermediate means subdivision, suborder, subfamily, subgenus, variety, subspecies, or clone, pharma, etc. It can be used or not used. That is intermediate. It is called. This is the hierarchy, how it is there. It should be sequential, it should be followed either in a decreasing or increasing way. Kingdom, division or phylum, because usually we use division only, but zoologists they follow phylum, class, order, family, tribe, genus, everything. This is there. Of course, here we are seeing means this tribe is missing here. Here, whatever Wikipedia or whatever you are saying means between family and genus, we plant taxonomies. We use the tribe between the family and this tribe and subtribe we use usually. How to remember very easily this thing, kingdom to species, how order. Kings play chess on finely grind sand. That means K for kingdom, P for phylum, C for class, O for order, like that, and genus and species. But here tribe also to be, that is why I have proposed like this. Kindly do come over for taking ginger soup. This, if you remember, means you can easily, the sequence you can remember. Kindly do come over for taking ginger soup. Just here I explained already, we are 15 minutes late running. That's why I'm not going to explain much in this. This is there. Of course, order is a group of families called this. Suppose Lamiales means Lamiaceae, Verbinaceae, Avicennaceae. This plantain, these things are included. That family means group of genera are included in this. Genus means group of species or intraspecific tags. Of course, you know all about the species. This is there. The rules of nomenclature prescribe the categories into which the plant should be classified. The categories constitute the units of classification. Their sequence and order is important, is fixed by the rules only. Change in the sequence is not allowed. The units of classification are arranged rigorously in a descending order. This I have taken from the book, how this Davis and Haywood book I have taken. Similarly, you can see this Lawrence book I have taken. This is published before 1950. There you can see clone word was used. Although we are knowing this clone term after 1980 only, that sheep was bred. But this clone word was before 1950 itself, it was existence. And certain rules are there how the ending should be there. This name should be end. What is kingdom here? They have given division, Thalophyta, this Bryophyta, all phyta endings are there, phylums. This class should be end always with the ONEAE, that should be end. Similarly, subclass should be ended as ADA, it should be end. Order should always end with ALES, Lamiales, Rosales, like that, Myrtles, like that it should. Order, suborder always should end with INEA. It should be end. Family, as you all know, ending with CA, of course, 8 families you are knowing, that is exempted, that is conserved names are there, like Labiatae, Cruciferae, Graminae, like that 8 names are there. This subfamily, this CAE ending for the family, this subfamily always it should be end with the OIDA. This tribe should be end always with EAA, like Roseae, Menthiae, Lemie, like that. Subtribe should be always end with INEA. This you should remember always how the ending should be there, like Roseae, Menthiae, like that. This, of course, our genus, everything Latin name would say, it is, I need not to explain that. And I come to the next topic. Of course, this most important topic is typification of plant names. I will explain very simple way what is holotype, this thing like that. Of course, I have given this photo. Why? Because, yes, all you know, for search of Sanjeevani, he has gone, he could not identify. The Hanumanji, he could not. Incidentally, today is Hanuman Jayanti. This he could not identify. That is an entire hill, I mean small hillock itself he has brought to this Sri Lanka. For that purpose, to cure this Raman and his brothers, it is there. Same thing for any taxonomic work, this experiment or any kind of experiment work, identification is important. How Hanumanji he could not identify the plant, like that we cannot do experiment everything. We should know the name of the plant correctly, how it can be, name of the plant can be correctly based on the type only. Otherwise, lot of confusions will come and it will mislead everything. What is a type? General term. What is that? These are the specimens or illustrated to which the name of the taxon is permanently attached. While publishing the new name, what specimen is used, that is called type. In other ways, the type, those specimens are illustrated connected with the name of the taxon permanently when it was first described. That is why type is called nomenclature type. Type is an integral part of the plant nomenclature. Thus, type will help us to know to which plant name it is actually attached. Simply as this, it is a very important to remember that a name has the type, but the genus species, they do not have type. Of course, we are calling as type species of this genus like that, but that is a type only we have to call. For the name only it is attached. If I say there are type specimens of 15,000 species at Are you hearing? Hello?
Yes,
Hello?
sir.
Yes. Because it has come unmute like that, it has come. That's it. See, if I say there are this 15,000 species are type specimen species at this, it is wrong. We should say always there are type specimens, these many names, types specimens are available at Central National Herbarium. See, I think, yeah. If one writes Barginia is type species, Barginia divari, it is incorrect. It should be written Barginia type is Barginia divaricata for that genus. I think something scratches has come. I think correct it. I think you can see the slide, I think. Hello?
Yes, sir, we can see.
Yes, sir.
I don't know how it has come, the scratches suddenly. This mouse moving, sometimes it happened. In every slide it has come. See, what is protologue? A protologue is nothing but where first the species was published, where that has been validly published. That is called the protologue. Suppose in some journal, you see Bulletin of the Botanical Survey of India, or now it is called Nilambu, that has been published first means that is called protologue. In this page number that has been in that volume first is published, that is called protologue. What is holotype? Holotype of a specimen, a species. I think moving mouse means it is giving something. I think once I escape and I will try. Sorry for this thing. I think you can see. I think.
Somebody's
Therefore,
fiddling on the screen.
species are intraspecific taxa.
Sir, please share one.
Pardon? I think this art. I don't know how it has come. By moving out the mouse, it is happening. I don't know. In Article 8.2 says, for the purpose of typification, a specimen is a gathering or part of a gathering of a single species or intraspecific taxon made at one time. If you are going to describe a new taxon and cite two gatherings as type, it is invalid. Why I am telling? See, sorry. Ajay, it is disabled by participant screen sharing. It is showing. Are you seeing now?
Yes,
Yeah.
sir. It's been presented, sir.
See, suppose you must cite one of them as holotype. We have to compulsory. See, for example, in 2011, Cytadian Jeopardy was published with two specimens. One is collected in January 2008 with a number 149, and the another in 29 with a number 339. That is why it is not validly published as per the article 40.6. See here, holotype already I told, when you are describing one specimen should be considered as a holotype. Here isotype is the duplicate of the holotype is called isotype. It can be more than one specimen also. See here, Feneria nigobarica has been published by Balakrishnan and Totatiri. They have cited 3043 as a holotype and B2C, two specimen as the isotype. Both are deposited in CUL, Central National Herbarium. It has been deposited. Syntype, when they are describing this specimen, they have not mentioned about holotype or isotype. Simply they have mentioned more than one specimen or one gathering. It is called syntype. There I have told that same number of this, one is coined as holotype and duplicates of them coined as isotype. Here, more than one specimen collected from West Manipur or collected by CB Clerk are mentioned. These are called syntype. Paratype is that when holotype and isotypes are mentioned, other than that, other collections are mentioned. These are called paratype. But in the syntype, there is no holotype or isotypes are mentioned. Here, holotype, isotypes are mentioned, and extra specimens also mentioned. This is called paratype. And lectotype is when syntype, like what in West Manipur, Clerk has cited three specimens, three gatherings he has cited. Out of them, one we can select, that is called lectotype. That is called lectotype. And neotype is there when original materials, that is whatever holotype, isotype or syntype, all are destroyed due to some or other reason. It destroyed, not available. In that case, one new neotype can be most probably from the type locality itself, it can be collected and it can be designated as a neotype, or illustrated and connected with that also. It can be mentioned as neotype. Epitype. These are the main types of types only I am covering. Sometimes in the holotype, lectotype or neotype, never serve for the, suppose some Ares specimen, they degrade very quickly. That is why we can epitype. Above that one specimen, we can choose it as a epitype. That is, epi means above or on above, but it should be affected when holo, lecto or neotype should be present. That is, I think epitype concept might be somewhat confusion to each and even for taxonomy is also confusion, but when it cannot serve as a type for no longer because the specimens are poor or it is somewhat destroyed or like that means only epithet can be coined with the other specimen it can be coined. The same thing only I had this. Here, my colleagues Bandavati and Patacharya, they have proposed that in taxon, in 64th volume, they have proposed some 5 years back. Epitype is a specimen must be selected, serve as an interpretive type when the holotype, lectotype, or previous designated neotype of a valid published name is demonstrably ambiguous and cannot be critically identified for purpose of the precise application of the name to a taxon. They have proposed in the taxon and it is accepted also. Now I come to the other topic, type. Only different types I have covered: holotype, isotype, syntype, lectotype, and epitype I have covered, including paratype also. Now I have the rule of priority I will give. In different parts of the world, same plant name usually develops in different names because knowingly or unknowingly or sometimes they think these are different. That is why obviously several names are based. For example, I am telling for some of the species of mentha throughout the world, it is having more than 800 names they are having. Its index sequences, everything it is listed out. Before in the 19th century itself, for mentha alone itself, for particular species of mentha, more than mentha largifer, more than 800 names are available. It is important which one of the names should be selected for the taxon. We have to this. There, principles of priority it is applied. What is in the cases of family and genus, now for species, exemptions are permitted in cases of concern. Now, family name, genus name, and species name also, including species name also, names can be considered if it is widely used. Suppose a Terminalia alba, very well known. It cannot be Terminalia Arjuna is well known. Suppose it is changed to Terminalia alba, it makes a confusion. That is why Terminalia Arjuna name can be considered. For example, here Mentha spicata and this Mentha spicata 1753 itself species plantara lineage published. But in the same place itself, after 1 or 2 pages, couple of pages, Mentha crispa, same species also, more or less same with a minor variation. Yes, coined for this Mentha spicata also. Same way, Mentha vidis also, Mentha spicata variety vidis also, yes, published. 1753 itself, in the same volume with a page differences, yes, published. That vidis transferred to 1763. Same thing, here, these are the synonyms, all are same only. You can see it. This opis first in 1825 published as a Mentha Walteriana. And after next year, same Mentha spicata he published as a Mentha tiniflora. Same thing, Wildinova, in the same volume itself, more or less in the same page itself, Mentha levigata and Mentha atulata, yes, published as different species with minor variations only. Now, after revisionary studies, we find Mentha spicata, everything will come. See, everyone was commonly, it was known as Mentha pudina only, it was known. It is there. But this correct name is Mentha spicata. What we market is available pudina. That is there. It was known in India, Buchanan and Hamilton coined the names Mentha pudina. It was validated by the Bentham in Plantarum Asiatic. Mentha pudina is nothing but a Mentha spicata, spearmint. What we commonly call as this pudina. This sorry. The rule of priority cannot be applied for the names in case of Permotsa and Tedaway. It is applicable from the 1753. Names published before this date need not be considered. With the rule of priority in the first half of the last century, that means between 1900 and 1912, a lot of nomenclature changes were happened due to this rule of priority. And many of the non-taxonomies like physiologists, genetists, including foresters, they were very much annoyed with the taxonomies. Why you are changing? Still, Still that many non-taxonomists are annoying with the taxonomy. Why you are frequently changing the even cytotaxonomists they were annoyed about that. Of course, presently provisions are there to conserve the species name. After Tokyo Code, we can we can conserve the species name also. Here, that after priority, I come to the effective and publication. What is effective publication and value effectively published and value? The name must be published in a medium appropriate to the requirements of the code. That means you cannot publish it in a local newspaper. It should be in a ISBN book or in the ISSN known journals only, well circulated journal only. And the name should be published in accordance with the provisions of the code. Whatever the code it is telling, according to the provisions only, you should be published. Suppose a Latin name only should be published. That is one this. Then valid word. One of the conditions of the valid publication should be it should be effectively published. Effectively published means it should be very simply telling means it should be well-known journal or well-circulated journal. It should be published. You cannot publish it in a local souvenir. You cannot publish it. That is. And Naman Nodam without plant. Naman Nodam is not effectively because no descriptions are involved. That is why it is not or no diagnostic characters is given means it is called as Naman Nodam. Names of families and lower taxa valid only if the nomenclature type is indicated on or on after January 1, 1958. This is the effective and valid publication. This. And the new names and combination published on after January 1958 are valid only if it is indicated clear the rank of a taxa. It should be followed by genera nova, species nova, tribe nova, variety nova, or combination nova. This should be accounted. New combinations are valid of the other does it in the original publication. Mere incidental mention of the name will not suffice. On or after January 1953, Bayesianum should have been clearly mentioned. Previously, they are not mentioned, but author's name they are indicating means we are. But after January 1953, Bayesianum should be clearly mentioned. The name of the taxon of the rank of the family or below is valid only when the nomenclature type is indicated. Such names, however, can be validated by subsequent authors by typifying them. Suppose syntax it is published. That can be designated as lectotypic. Linnaeus had never indicated the type materials. However, many of these names were lectotyped by various authors, mainly by Jarvis E.S. Mead. And I am coming last but fifth one, author citation. It is the most important one. But of course, very briefly only covered. I am not going elaborately because for college or college students, when workshop is author citation itself, 1 or 2 hours classes I take. I took already in Calcutta. But here, very briefly, I will go. Before the Linnaean, the botanists used to clarify the plant names by citing their original author after the name them. Before Linnaeus itself, they have started to mention that. Historically, the principal need of updating the author names to give conveniently short way of indicating a name. Suppose L dot itself indicate this is Linnaeus. Not only this abbreviate, it is standard form of the author's name because it saves lot of space, printing material, everything, cost of printing, everything we can save it. For that only mainly we are using. Suppose everywhere we are writing Linnaeus or J.D. Hooker or Jackson Hooker, we are writing means it's occupy the space. That is why standard form using is enough. Decandle also, where it occupies a very big name. Risenberg, it will occupy very big name. The international code now contains sections which allow for formal citation authors names. In the book, the authors of plant names by Brummett and Powell, they have nearly 30,000 standard form of the names they have given. They refrain from using the other abbreviation. Of course, we commonly they call it as other abbreviation. They call it as a standard form of the other. Once the standard form of the name is fixed, it cannot be changed. So here, Begonia scandens, 3 types there, sir. But this L only indicates Linnaeus correct name. Why I understand? This Lin is another one. Lin Manson Bruce is the abbreviation for Lin. For Carl Linnaeus, L itself sufficient. That otherwise, what another Linnaeus given is full form I have given. Actually, we should do for Linnaeus, L should be used. For Australian Manson Bruce Lin, it should be Lin should be used. And this is a sample of the page I given from Brahmit and Powell book. Just I scrolled that. One will understand is how they have abbreviated these names. I mean, abbreviated standard form they have given. See, Aren't whole cities there. They should have put Aren itself. AREN itself they would have put. Aren's full name, of course, they have given. Still more clear means here, Armitage means arm they have put. Why? They should have put here army they would have put, or they would have put AR itself they would have put. Why? Armstrong. They have put ARM, STR, full stop they put. They should have put in arms itself, they should have put. Why? The rule says that always the standard form should be made between consonant and vowels only. Here Armstrong it is there means here O is coming, it is vowel, it is coming, before that they have in that. Here, of course, initials I have given, that is CA, everything. Always the Armstrong, John Francis already he occupied, that is why they are given the initials before that. Otherwise, confusion will. Suppose my name is Sambat Kumar, I usually abbreviate as VS Kumar. Suppose all are started to use V Santosh Kumar is there means he can also use VS Kumar. That is why to avoid the confusion only, some extra initials they have used. That we will see here. Here I have given some standard ones I have given. This Armstrong, everything. This Bandavadhyay, it is abbreviated Bandavadhyay. Bentham, Benth they have given. You see here between consonant and vowel they have abbreviated. This Benth they have fixed. Of course, Cook, already other Cook is there. That is why Theodore Cook T abbreviation they made. Here, Father William Jackson Hooker, it is there, his father name. Here, they have not adopted Hooker, J.D. Hooker. Instead of that, his son, Phileas, they have adopted. Here, Gamble, he used full name. Dalzell, they use, as far as possible, full name can be used, but provided it should be usually less than 7 characters. In code says maximum 10 character it can be allowed. Not code. In that book they recommended that Brahmit and Powell less than 10 characters using is the best, and it should be better between 5 and 7 is the best. My name is Kumar Sambat Kumar Venkatachalam. It is abbreviated as VS Kumar. This is internationally accepted. Similarly, Sanjapa's names are also, it is internationally accepted as M. Sanjapa. The standard form or name is surname abbreviation. It is sometimes it D Condol is there. DC they are accepted. Suppose his sons are there means A Condol or Primate Condol means BDC, like that it is there. Even this Linnaeus name also abbreviated with a single letter. And with or without initials also it is because Phileas it has been used. And another one thing I am telling, all abbreviations, it should be always, its contradiction should be with the full stop, it should be compulsory, it should be made. Of course, conic and this conic are love. This apostrophe, this Ogara, Japanese name, or these are treated as homonyms, same name also. It can be used as conic also, it can be used as, in English, conic also, or German language like conic also, it can be used. And whatever in Brahmit and Powell book, maximum they have followed in taxonomic literature to whatever standard form they use it. Of course, some deviations also they made. Here I have given the what is taxonomic literature. George King, the first director of Botanical Survey of India, that his name is King, George King, abbreviated as King in the full name. Here, same thing and I am explaining. That is surname Hooker because Phileas it has been used. Of course, if it is more than one persons are there, what do you say for Robert Brown? Here 2 or 3 Robert Browns are there. It can be with the suffixes, it can be used. First Robert Brown, father is called R.Br. After that, Bis and his grandson is tertiary. Bis and TR also can be used for Robert Browns. How to cite the other abbreviation? This a simple example from my thesis I took and gave. Because Archimedes, this Bentham, he has first published the Archimedes subfamily in Labiata genera species, his own book. That is why we are not used any in or anything, simply comma we use. Here, after that, in decandol also he used the same name. That is why in decandol prodromas I have mentioned. And Bentham, in Genera Plantarum, this book was jointly written by Bentham and Hooker. That is why I used the yin. Here, usually, Hooker means Hooker comma, Flora, British India. Hooker in Hooker we are giving means it shows that in this book, more than one author is involved in the Flora, British India. When journal name is used, there also we use Mukherjee in records botanica. Of course, nowadays, everywhere commonly they give it as comma. They are not using in many journals nowadays. They are following simply comma. They are following. That is why in DC or in Bentham and Hooker or CB Clark in Hooker up like that they are not giving because to save space to save the confusion also they are following like that. But this is the standard way of giving when the author is published in someone's work, in should be made. Someone's work or in journal, in should be used. The same thing, this Elsewhere major just to show that here, so I have made this combination in status nova I made because Elsewhere Stachydas variety major was there. I have found the flowering material because Hooker published this major variety only based on the fruiting material. When I have elaborated the description, flowering material I found from the Dehradun, then I made into the species rank. That is why Elsewhere major Hooker, V.S. Kumar and B.D. Sharma, yes, combination and status nova. I made combination also to the species rank, plus its status has been changed to this thing. And another one thing you have to see here, many persons they use senso and octagon, some confusion. Suppose only one citation only you are using, it is better to use senso. If more than one author, one literature you are citing, better to use instead of senso, octagon, Bentham and year. This is the way to cite. Of course, these are the PhD students who are doing, they only do. Same thing, this masala also, same example I have given. This thing, of course, I have more or less I have made, given everywhere. And with this, come, I think only 10 more minutes only it is there. Last part. These are the nomenclature notes on few flowering plants of India. I have published in the phytodiversity. This some of the examples I will show. In this Gentianaceae, I have made Naman Nova, new name I have proposed, Gencinella berkeley. Why it has been proposed? Because this Presexia, we have found that this is better fitted to the genus Gencinella. That is why we wanted to transfer. Actually, when we are transferring means specific epithet should be retained. Unfortunately, already in 2008, this Gencinella suminthi was published for the some South American plant. It has been published. That is why we have coined the new name Berkeley, because this Berkeley has published this species. New name we have given because it was preoccupied. That is why we have given a new name. Similarly, here, what combination now I made? Actually, rubus irritans variety sub enormous new combination because rubus purpureus, this species was transferred to the irritants, but this variety was not transferred. When we found that, then we are transferred to the rubus irritans, that is when new combination has been made. Similarly, here also we have made new combination because this Bonias limnophilides we transferred to the genus Limnophila. Finally, I am coming to what is illegitimate names. There, how to cite the author's names. I have given author standard abbreviation, how to cite correct author's name, and literature how to be cited. Here, I am coming to the final part. What is illegitimate names? So far, we are given legitimate about the legitimate how to publish validly, effectively. Like that, we have seen. Here, what are the illegitimate? A illegitimate name or epithet is one that accordance with the code or provision, it is called, according to the code, it is all the provisions, it is all the articles it is satisfied means it is called legitimate name. Those are contrary to that called illegitimate. Suppose I told, suppose you are publishing in the local newspaper or it is not, that type is not cited correctly. That is a holotype or isotype. After 1990, holotype names should be used, but without mentioning that you are publishing it, that become illegitimate. What is superfluous name? If it is the same type material used for publishing the other, because Povastamon already one name is available, but this Rapid has published as Australis. After that, raised in 1970s, Anuragi also based on the same type material they have published. But when Povastamon name is available, they should not have published the new genus Australis or Anuragia. This later homonyms. It is possible later homonyms. Suppose I have published some name, Rubus sanjapi I have published. For other species also, I have published from the Western Ghats. Someone has collected a more or less similar time he has collected. Someone from Andaman also, some Rubus specimen someone has collected. And suppose he is also coining the name for our former director, Rubus sanjapi means that became a later homonym. And of course, I have told that Naman what is Naman Nodam? Naman Nodam without valid description or any diagnostic character, it is published in Naman Nodam. Best example is Roxboros, Hortus becalensis is the best. Tata names are not allowed in botanical nomenclature. Of course, in zoological nomenclature, Tata names are allowed. Here, Malus and Malus is a Tata name. But Gajanus Gajan is and Suspenia Suspan are not Tata names, not Tata names. Anyway, before concluding that there are lot of codes of botanical nomenclatures are there. Of course, we are calling it as ICBN or ICN. We are calling. There are other things. Are international code for botanical nomenclature of cultivated plants, that is abbreviated ICNCB. International code of zoological nomenclature is there. International code of nomenclature bacteria are there. International committee for taxonomy of viruses are there. There are a lot of difference between our botanical nomenclature and zoological nomenclature also. Of course, I am not going to go further because only two difference. In 1753 onwards only our 1753 all names are validly published in as per our botanical nomenclature or our plant nomenclature. Zoological nomenclature from 1758 onwards only it is valid, and Tata names are not allowed in botanical nomenclature. In zoological nomenclature, Tata names are allowed. I think I have covered in within 1 hour. I have covered. Thank you very much. Thank you very much.
Thank you for the lectures. Now the participants can clarify their doubts.
Sir, there are two questions in the chat box, sir. Shall I ask?
Shall I close my sharing, or maybe useful also? Please, madam.
Yes, sir.
Because I am not getting chat box. You please tell me.
Yes, sir. I'll read it, sir. Sir, the participant is asking, can you add a note on APG classification, sir?
Yeah, that is a. Anyway, I will tell. Sanjapa sir will elaborate because he is my mentor, my supervisor. He is the best person to tell. Of course, simple one word. Angiosperm phylogenetic group. It was formed before 2000 itself. It was formed around 1998. They followed. Now it is the fourth classification. It has come. APG1, APG2, APG3, 4. Of course, APG from APG4, there won't be much difference won't be there because all phylogenetic aspect they have done, all molecular materials they have used. That is why genetic material used. That is why we feel that there won't be a drastic change in the APG4 classification. Of course, from APG2 to APG3, APG4, lot of contrasting classifications are there. You are astonished to know that most of this Crapulariaceae and Orobingaceae, these are splitted now. Most of the Crapulariaceae has gone to Plantagenaceae. And what are the parasites in Crapulariaceae that has been transferred to Orobingaceae? Myself itself, now we have to, each and every time I have to check plants of the old online, I have to check which family it belongs. Even my own labiata itself, from Mirbidaceae, from 8 genera of Indian 8 genera. I don't know how much, nearly 12 genera throughout the world transferred to labiata. Even Tectona grandis, well-known Verbinaceae member, or Calicarpa, now they are in labiata. This is, I hand out to my mentor, Sanjapa sir, to explain about the APG4.
This is basically a question regarding classification. So classification, I saw on Makram Daitwade to asking another question also, which classification should be followed. So it doesn't matter which classification you follow, it should help you to understand the relationship between the different families and their evolutionary sequence. So APG system is being done based on a few gene loci that have been used in the phylogenetic system, phylogenetic part, particularly for the monophyly and the polyphyly thing. And many families have been displaced here and there. But if you ask me, as an old taxonomist, I would prefer Bentham-Underker system because it's very easy to understand the relationships of the families, at the same time identification. So what is the purpose ultimately? It is the identification of the plant, which is very, very important. But if you look at the phylogeny, and maybe this APG system is important. But again, now this APG system is based on a few gene loci only. Tomorrow, when the entire genome is mapped, this classification is going to really change in a very drastic way. So that we have to wait and see. And as Sampath Kumar said, APG4 and maybe in another APG5 also comes, there may not be much changes in the system of classification. So the system of classification and nomenclature are two different things. Don't mix up these things. And for the time being, we should follow for easy identification. If you ask me, Bentham-Underker system is the best. Yes, could you understand, madam? The person who asked the question? You have any other?
Sir, yes, am I audible, sir?
Oh, yeah.
At that, sir, is the sir said that Malus is used for tautonym, but Cajanus and Sesbani are not used. Why this is so?
No, Malus is also tautonym. Cajanus is also tautonym. They are not accepted.
Okay, but why? Yes, why this is so?
No, because the specific specific epithet should not repeat the generic epithet. This is what is provided in the code. Whereas if you go to zoological nomenclature, there it is, say for cobra, it is najas. So there they have permitted the use of the tautonyms, and in botanical nomenclature we don't accept the tautonyms. Repetition of the names.
Okay, sir. Okay. Thank you, sir. Thank you.
Any other doubt, you tell me. If you don't, you didn't like the answer, means you should question. You can ask.
Still,
You should clarify.
Yes, still some doubt, because malus is tautonym and others are also tautonym. But why we are not accepting the remaining two?
Sesbania, Sesban. It is not Sesbania. Sesbania, Sesban. That is accepted. Kejanus, Kejan. That is accepted. Not Kejanus, Kejanus. Malus.
Okay.
You understand?
Okay. Yes. Now clear, sir. Okay. Thank you. Yes. Okay. Thank you, sir.
Okay. Nomenclature is a thing one has to practice with whenever you come across a problem. If you practice and then read back into the code, you understand it much more clearer. Otherwise, we will have to give examples for every every aspect of the code. As we said, what is a holotype, what is an isotype. All these things with examples only you understand much better. Of course, he has given an overview of the code and at least a few most important applications in the code.
Thank you, sir. So I guess all the participants have clarified their doubts. By this, the first day of virtual lecture workshop comes to the end. Henry Ford said, "Enthusiasm is the yeast that makes your hopes to rise to the stars." With these lines, I conclude and expect all the participants to join us tomorrow with the same enthusiasm for the second day of the virtual lecture workshop. Thank you all.
Thank you. Okay, Sampath, okay, fine.
Thank you so much, sir.
Thank you, sir.
Thank you, Sampath Kumar ji. Thank you, sir.
Thank you, sir.
No, Yadav Sahib is still there.
No, I am listening whole session. I was there.
Okay, you come up tomorrow again.
Yeah, I will be there.
Yeah. Okay.
Okay.